علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان لا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Jazakallah. Ladies and gentlemen, let me introduce our speaker, Professor Dr. Hafiz, to you. Professor Dr. Hafizur Rahman is working as a professor at Dhaka International University, Bangladesh. He has more than 61 publications in different international peer-reviewed journals. He has received mixed fellowship from Japan for his PhD and has been awarded several research grants. And in addition to this, he has long association with International Center for Chemical and Biological Sciences, University of Karachi, where he has successfully established a molecular diabetology lab and has expanded his expertise in the development of diabetic models in rats and used these models for different research projects. He has trained a number of researchers from Bangladesh, from Pakistan, and also other countries in the field of anti-diabetic drug discovery. And in addition to this, Professor Dr. Hafiz is also comstech's distinguished scholar from Bangladesh. Sir, extremely grateful for your presence here. And I hope that this workshop will be of valuable information to the participants who are attending this. And on behalf of Comstech and on behalf of Professor Dr. Iqbal, I'm extremely grateful for you, for your time and for your presence here. With this, sir, I request you to please start this workshop with your opening remarks.
Bismillahirrahmanirrahim. Thank you, Kazima, for the good introduction, nice introduction. Dear my dear brothers, sisters, and friends from different institutes of Pakistan. It's an honor for me to be here. So, as Kazima told, I have a long association with IGCBS, University of Karachi. I have also long association with IGCBS. I welcome most of you. I welcome all of you to our workshop, Drug Design and Development, the Art and Science. So, because this is Muslim Ummah, after pandemic, we can understand if you have money, you can buy anything. But if you have money, you can buy the medicine. The medicine or drug is developed in Western countries mostly, and it is developed for their needs on accounts. In WHO, remarks that the BMI of Asian population is less compared to the Western population. The Western population is after 25, but here 23 is the obese. So BMI marker is different for Asian people as well as non-Asian people. However, in Asian people, Muslim people, BMI is different, our pathophysiology is different, our food is different, our culture is different. But medicine is the same. So one medicine may save one life, but it kills others' life. So one size doesn't fit all. If one size fits all, it fits no one. So it is the time to think ourselves. It is the time to think seriously and honestly how we can develop our own medicine, how we can make our own medicine demanding our own population. So hope and drug development procedure is a long procedure, risky game, long time. And it needs people from chemistry, biology, pharmacology, statistics, a teamwork. Alone cannot do it. And it needs industry and academy collaboration, cooperation. Hope in this respect, YC play an important role for development of drug for the Asian people as well as for Muslim peoples. So comstech play an important role and comstech play an lead role, and we of the peoples, you can think it. If you think today, it will take time. It will take time. At least if you think, if you can start now, it will take time. At least this is the time we have to think. So it is the time to think seriously, honestly, and properly. How we can manage our life, how I can, the Asian people or Muslim people, we can make healthier life, happy life with medicine or no medicine or with our own medicine, so that our country will progress and that will be Muslim Ummah for the betterment of the happy and with the future of our Muslim Ummah. With these few words, I welcome all of you. I hope last 3 days I'll be with you. So rather than giving my lectures, mostly we'll do interaction. I will share my experience, expertise. I am not expert. I know something, what I did by myself. Okay, I just will share my knowledge. By sharing knowledge, hope we can ignite your inspiration, we can motivate you so that near future we can work together. We can work with teamwork, with the connection, with the cooperation of comstech, and it will help Pakistan, it will help the Muslim Ummah for the betterment, and for the betterment of Muslim Ummah. With these few words, I thank you all. I will come out my lectures. Thank you very much. Thank you.
Thank you very much, sir. With this, we will begin the very first technical session of our workshop. The title of this technical session is Importance of Drug Research, Strategy and Drug Development, Ethical Consideration and the Clinical Trials. As we know that drug research and development is very important for the countries, for the social and economic benefit as well. And I hope that in this technical session, you will get to learn that what are the strategies and what are the ethical consideration, very important of them, that we need to consider during the drug development process. So I request you to please start the technical session.
So it is only also in online?
Yeah.
Okay. Okay. So can I see the online participant?
Yes.
So before going to my main lecture, please, my participant, please, can you introduce one by one?
We can start from here.
Yes,
I think. Okay, sir, I request you to please introduce yourself.
My name is Sayyidul Rahman. I'm a pharmacist. I'm working in Emerson Vaccines and I'm a production manager.
Thank you, sir, for coming, sir.
So, my name is Murad Alam. I'm a pharmacist working as a R&D minister and some pharma.
Sir, thank you for coming.
What is it, currently to learn our PhD in biosciences. Now I'm looking for the opportunities.
Okay, thank you for coming, sir.
Offered this one, final stage PhD student, and I'm also working in thanks to the
Okay, could you please mention your institution as well?
concepts.
Concept. Sir.
Sadar from Federal University is located in microbiology and my research is on
Okay, madam. Thank you. Thank you.
now wonderful from Federal University. I'm Mohan Jawad and I'm from Dutch. I'm doing my MS in industrial biotechnology and I'm working on microbiology.
And you're also doing your MPhil?
And
MSO.
my name is Rajan Sen. I'm working as an instructor at the Department of Biosciences for Community University staff.
Thank you for coming.
I'm Abdul Alim and doing the job AP and Epidemiology Scientific.
Sir, thank you for coming.
I'm Zubair, PhD student from Biotechnology Department, Qadir University.
Okay, thank you for coming.
My name is Mohammad Usman. I did my MS Molecular Biology from Comstech Islamabad, and now I'm applying for different jobs.
Okay, thank you.
I am doing my MPhil from Computer University in microbiology and also working in Japan International Hospital as a microbiologist.
Okay, thank you for coming.
I am Mohammad Sabdar and I'm an investor by
I can introduce gentleman next to him. He's Professor Dr. Javed Khurshid. And Dr. Javed Khurshid is working as a consultant in the area of science communication and diplomacy. So thank you very much for coming. It is a pleasure to have you, but do say a few words if you would like to.
Come. You're supposed to come.
Thank you so much. It's really a pleasure to come here.
So you have your things. I thank you for the address the audience as well.
Thank you so much for inviting me to give some comments, and it's my pleasure. You have already introduced Dr. Moizur Rahman, who is going to lead this workshop, and it is my privilege to say a few words. Drug discovery and design is very important if you really want to get new and beneficial drugs. So instead of wasting a lot of time just doing the research, which was old method, now this drug designing will help us to find out the new drugs in a quick way and very perfectly. So this is a very important topic, and I see the pharmacists and the people from microbiology and biochemistry department all are here. So they work together for the drug design. And I'm really privileged to see Dr. Muiz guiding these students and these professionals who are involved in that. And I thank him for the future lectures too, if I'm not here. Thank you so much.
Sir, thank you very much. Sir.
My name is Danish Hassan. I'm doing PhD from National Institute of Biotechnology and Genetic Engineering, and my research area is relevant to drug delivery and enzymes.
So are you doing it from Festival?
Yes.
Yes. Okay, thank you for coming.
My name is Yaz, and I have done my MS in biochemistry and molecular biology from Comstech University, and right now I'm working as an assistant lecturer in my university.
Okay, thank you for coming.
My name is Guru. I did my master's in health biotechnology from Faculty of Bahashuli University, and my field is related to this project. So I have performed my work in this field, and now I'm also working as a lecturer.
Okay, thank you for coming.
My name is Anisa Deep. I'm a student at University.
Thank you. Thank you for coming.
My name is Ritika. I am doing my chemistry from Academy.
Thank you.
Myself, Amna Sufyan, and I am currently enrolled in MS Organic Chemistry at Kaidan.
Thank you.
I am now the research student in organic chemistry and other things.
My friend.
Yeah, I'm even Malik and I'm officer at comstech.
Thank you for coming, both of you.
My name is Sayyid Manisha and I'm currently studying in doctor of pharmacy from Ipada University.
Thank you for coming.
My name is Aviskar and I was also enrolled in Chicago Medical University and I can be one of them.
Thank you.
My name is Mohammed Hussain. I'm a pharmacist and from Chicago Pharmaceuticals.
Thank you.
Dr. Khalid Mahmood, I'm associate professor and pharmacist, basically PhD in microbiology and serving in active administration.
Sir, thank you very much for coming. So sir, you see that we have many people here from diverse backgrounds and also from different institutions. So over to you now.
Thank you for introducing yourself. It is good to know that it's perfect, because the drug design and discovery needs multiple people. Chemistry is organic chemistry is there, biochemistry is there, biochemistry molecular is there, biotechnology is there, microbiology is there. So, and we all are a statistician. Okay, that means it's perfect match. Thank you, Kazma, for arranging from different background, from different university. It's perfect. So, I'll share some of my knowledge. The best way, so you people here join here, just make friends by collaboration, cooperation. You can 1 + 1 sometimes make friends, make group. Chemist, biochemist, microbiologist, molecular biologist, make a group. So you can make a group, WhatsApp group or something to Kazma will help you. So possible to make a WhatsApp group, then we can share all the slides, all the materials to them.
Sure.
So without any delay, I'm start my lectures, drug design and development, the arts and science. Okay. No one. So drug design and development, what is art and what is science? Let's see. So welcome to this workshop. So already Kazima discussed and elaborate what we will study in this workshop. Drug design and development, the arts and science, biotechnology and drug discovery, from whence to be site, development of proper animal model, and how to translate in vitro to in vivo. And mostly for toxicological study, we have to do some histological work. And most important is the training of humanity. This is our main motto of this workshop. So thank you, Kazima, and thank you, comstech, for inviting me and give me the opportunity to share my experience, expertise with you. So as Kazima told, I'm from Bangladesh, okay, and my hometown is very nearby India, border side. My hometown and India only 8 kilometers. And most important thing is, it is the unique place in the world. It is the unique place in the world. That means inside Bangladesh, some Indian territories there. And inside India, there is some Bangladesh territories there. You see the map, this India, but Bangladesh is there. Some land is there. And it is Bangladesh, some Indian land is there. It is very unique. Okay. So previously, in the morning time, we have to go there, but before sunset, we have to come back. Okay. So few years back, the study resolved. We exchanged the technical territory. Okay. So still, some people in my hometown lives, okay, They lives in Bangladesh. They cook in India. There's the kitchen, half of the kitchen it is Bangladesh and half of the kitchen in India. Okay, this is an interesting story. Okay. So this is the University of Dhaka. I graduated from there from different biochemistry and molecular biology. And after that, I worked, we call it Bardin. This is the diabetes hospital in Dhaka, and it is the world largest diabetes hospital in the world. In a sense, every day, 3,500 patients visit here. So all over the world, there is hospital. And the hospital, there is a diabetes wings. But this is hospital only completely focused on diabetes. It's my pleasure. I work there for the diabetic patient. Then I did my PhD in molecular genetics in Kumamoto University, Japan. And still, I have a good connection. Still have a good connection with Japan. These students, researchers, with different background. If you want to do some collaborative research with Japanese professor in your field, I may help you if it is mass. Okay, sometimes once I did PhD from Japan, I'm a Max Fellow. I am in track 2. I have easy access to Japanese embassy at anywhere. Okay, so that if I write a recommendation letter, if you recommend you and write in PhD Japan, it helps. It helps. Take this advantage. Then I worked in Dr. Panjani Center for Molecular Medicine and Drug Research, HEG, University of Karachi. I worked there 13 years. I established molecular diabetes lab there, set up diabetes laboratory there. I worked 13 years. So I worked there for my golden age, golden time, active time, and most productive time. So due to COVID, just before COVID, I was in Dhaka and I started there. Okay, this is the beautiful building with beautiful people, beautiful mind. So then in Dhaka, I worked as a professor of Prime Minister University. Now I'm working as a professor and chairman, Department of Biochemistry and Molecular Biology. This university, we call it Dhaka International University. They are my colleagues and students. We are working together for the betterment of research and for the molecular biology. Okay, now come to the point. Drug discovery and development. The process are working. So any process, it takes time. It takes time. If you want to make a cook something at your home, cooking start from where? Anyone can tell me? Cooking, if you want to cook some biryani, it start from? No, it start from bazaar, market. First you have to purchase this one, not kitchen. Kitchen is the intermediate. Okay, same is true. A drug designing or drug getting a market today. So if food is ready in your table, that means it cooked in kitchen. It processed in different places. Okay, then you are taking food. Maybe it takes 10 to 20 minutes, 30 minutes. Managing the things, it takes more than 30 minutes, hours, days. Same is true. But the discovery of drugs, it takes time. It takes time, long time, lots of money. So, and why drug is needed? It's management of disease. So from management of disease, for the cure the disease, we need drug design and drug development, drug discovery. So we discussed little by little. The art and science of drug discovery. Okay, the science is how we are doing the experiment. This is the science, the result. And art is thinking ability, how we can design, how we can think it, how we can manipulate it. This kind of thinking is the art. Before doing experiment, my professor told me always in Japan, so do your experiment first here. First, do your experiment in brain. I found this very interesting to me. before doing in Japanese people, why they developed? They don't do experiment. First, they do experiment in the head, in brain, then they draw a flow chart. And what kind of expected result may be, they design it. And they give it to the person or the supervisor. Okay, before doing experiment, they told me, if you do without understanding anything, you got the result. If you got the good result, you couldn't understand how it happened, how did they happen, the interpretation. If you don't got the result, you repeat it. So if you repeat it, it will be a technician. If you repeat an experiment, it will be technician. If you don't repeat it, think it, and repeat it in different ways, A, B, C, D, different plans. So result didn't came. Why? And probable answer is A, B, C, D. And fourth answer is E and I don't know. So therefore, so doing experiment always is not good. Before doing experiment, do your experiment, either brain, then you can think it, then you can do the experiment. In this way, this is the art and science. Art is here, science is in the play. So drug development, there is many processes. Let's see one by one. Okay. So I'm telling you one thing, disease, disease. One, you know, everything has ordered. Everything is ordered. If you go, everything is smoothly recorded ordered. We came Karachi to Islamabad by year, everything is scheduled. Okay, but if schedule is some disturbance, it will make all the disorder. Getting the plane, coming here, everything will be disorder. So one is disorder and normally easiness, some comfortness. So if you are fine, you have a plan to attend my workshop, it is your easiness. You came willingly, okay, but in the morning time, you couldn't find it. Easiness, it means diseasiness. Disease is nothing, it's a diseasiness. If you have disease, you feel diseasiness. So this is. So if you feel diseasiness, you have to come back to easiness. So diseasiness is disease. Disease to easy stage, you need medicine. So medicine or drug, it doesn't mean the pharma drug. Medicine is different thing. We have a wrong concept. Medicine, no. If you walk, it may medicine. If you talk, sometimes we went somewhere, no, Urdu mein bolta hai, Maulvi or Peer. If you went there, by seeing them, by listening their words, you feel better. It's also medicine. It works. Some Maulvi, if you take one glass of water, it is medicine. So medicine doesn't mean if you go pharmacy, you go pharmacist or medicine shop, if you take medicine, no. And music, it's a medicine. Reciting Quran, it's also medicine. And most important is the best medicine is the walking, and it is absolutely free. Only one medicine is free: walking, 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 and walking. If you walk, your body will be fine, as well as your mind will be fine. It is the only medicine that is 100% free. Okay, now there is a difference. Medicines and drugs. There is a little difference. Medicine and drugs. All medicine is drugs, but not all the drugs are medicine. So paracetamol, it's a medicine or drug? Medicine. Anyone? Paracetamol, it's a drug or medicine? It is medicine. You are absolutely right. What is the dose of paracetamol? 500. So if you take paracetamol 1 + 1 + 1, it's medicine. If you take medicine 10 + 10 + 10, 30 + 30, it's drug. Got my point? So drug, it has no doses form. Okay, but medicine has a dosage form. Sometimes, so during the operation, so using people epinephrine, pethidine, people use it for the treatment, a specific dose. Same pethidine, if you take pethidine, you take in different in extra doses, it will be drug. Okay, so medicine has a specific doses point. Drug has no. So paracetamol, it's a if you use a standard drug, a standard doses, it is medicine. But if you take too much, it will be poison. That means it will be drug. So what is drug? Now, normally in medicine, though it is different, normally we call it drug. So sometimes unmarried and bachelor, any difference there? Unmarried and bachelor, difference or no difference? Usually no difference, but there is difference. Okay, usually there is no difference, but there is difference. Same is true, medicine and drug. Okay, but normally we call it drug. So medicine, it's chemical substances to treat the disease. A drug defined by the chemical agent, it may be the chemical agent or it can be a biological product. Initially, all the medicines are from the chemical source, chemicals, from chemical agent. But nowadays, in after 1980s, okay, many medicine from the biology, microorganism. So this chemical agent or biochemical agent or biological product, it may be came from plant, it may came from plant origin, it may came animal, it may be mineral, microorganism. So most of the drugs we are getting in pharmaceutical, this one is chemical agent, mostly. Either it is natural products or it is in synthetic product. And sometimes we put some animal sources. Best example is the insulin. It comes from pancreas of animal, either pork or calf. Okay. Antibiotics, so it came from fungi. So that means initially it is a single source, now it is the multiple source of drugs. It came from different sources. So many drugs used today, okay, it is used today, but it's known for hundreds and thousands of years. This includes epinephrine, caffeine, opium. This is used as a drug, so maybe 100 years. 100 years. Drug discovery start, the modern medicine start in 100 years back. So before 100 years, how people manage it? So nowadays, you have a smartphone, right? You have Google location, right? So we can easily constant. Someone told, okay, let's do this. So the 30 years back, when I was a student, okay, I think there I have letter with me. Okay, this university, this address. I took this address. Okay, no look, I don't know anything. At that time, without anything, I went there. In 2002, 2006, I came in contact. At that time, internet facility is very less. Okay, so at that time, people live there. You will communicate. Now you have a smartphone. Before a smartphone, you have button phone, right? Before button phone, you have a land phone. So very few people have land phone. So people communicate or not communicate. The life is stopped and life was going on. So life going on in circumstances. So drug discovery process start 100 years back. So before 100 years, how people they manage it? How? Somehow the knowledge. Okay, their knowledge, practical knowledge, folklore, the previous knowledge. So I have something wrong. So my father asked me, by my father or mother or grandfather, told me, please use this. This grandfather learned this from his grandfather, his grandfather to his grandfather, his grandfather. It is we call message translation by generation to generation. It used. So from drug discovery, the basic concept is very useful. Basic concept, and then because drug, it goes to clinical trial. After discovering the drug, it will go to clinical trial. But before discovering the drug, we are in clinical trial. Without discovering the drug, we use the clinical trial. Please use this, the edible part, this. Same is true during COVID. Once disease start, medicine came at least 2 years back, 2 years later. Within 2 years, what we did? People ask, please use this, antioxidant, this. If no medicine, there is an alternative. If people manage it, okay. So the therapeutic properties of these agents discovered empirically from folklore, traditional medicine, or by skin plants, animals, and microorganism and pharmacological activity. So ephedrine in China was 500 years before discovered the modern medicine. And sometimes drug was discovered in well-designed manner. Sometimes we call it accidental discovery. There is two accident. One is accidental discovery in accident, another is called sweet accident. If it is accident, but for good for you, we call it sweet accident. Many stories there. So you miss the train or miss the bus. Unfortunately, the same bus, the same train was get accident. It's called sweet accident for you. Okay, so few drugs like sweet accident. Best example is the penicillin, accidental drug. Okay, now I'm giving a very simple example. Oat saline, ORS in Pakistan available. So people, why are using oat saline or ORS? People are using, right, ORS. Why? For what purposes? For diarrhea. My question is, this ORS was developed in 1971 in Bangladesh. So diarrhea started in 1971 or before 1971, diarrhea was there. This diarrhea was there from the human first day one. So how to manage it? Remember very carefully. Very simple example. ORS. Okay. So ORS was developed 75 years back, 1971. Okay. So only 75 years back, but 2,500 years ago. Okay, if someone has diarrhea, how to manage it? They use rice water, coconut juice, carrot soup to manage it. Okay. In 1831, William Brooke treated cholera patient with IV fluids, reducing mortality. In 1971, Dr. Dilip, he treated diarrhea with this ORS, and mortality is 100% to 0.36%. Okay, so I'm telling this one. Okay, what happens normally, or before the ORS in China or in Africa, if they have some diarrhea, how to manage it? They use the rice. If you make the rice with extra water, we call it ma. We discarded it. What they did with this extra water after cooking, the extra water, they took it for the management of diarrhea. Later, it is tasteless. Then they add salt. Okay. Then they add salt and found that it is far better in taste as well as managing diabetes. Okay, that means ORS is in market, or it is discovered in 1971. And 2005 years back, ORS in different form. today, so I am taking this kind of water. So 20 or 30 years back or 25 years back, this kind of water is not available. Water is matka, okay, and the glass. Same water in same water in different formulas, but it's presentable. Medicine is nothing. Okay, so my father, my grandfather told use this medicine, this plant, this plant. So from that plant, the active ingredient isolated, purified, and put in the capsule. This is the medicine. It's not miracle. Okay. So idea, it makes simple and make it marketable, make it so that easy for their human better health and life. So sometime I called it ICDDRB, International Center for Diarrheal Disease Research in Bangladesh. Okay. In 1971, they did in the 1971, sometimes we call accidental discovery. Okay, normally initially this kind of ORS initially give in IV. In IV. So giving IV, it's not easy. You need some trained people to give IV. So it is another accidental discovery. So that time 1971, many people, lots of patients suffering from diarrhea in Indian border side. So it's not possible to give ORS, not possible to give injection. Some nurses give oral without knowing anything. Without knowing anything, they give orally. It works. And it's miracle. Extra water of rice, salt. And ORS, it's not same, but similar. In extra water of rice, it contains carbohydrate, it has sugar, and you added salt. So in ORS, it's nothing. Sugar, salt, in different ratio. Okay, it makes save the life. And for the ICGAB, they developed this drug, and for the betterment of the human life, save more life, they didn't be patent. If they make it patent, the Professor Dilip told, it will take 5 years to get patent. In 5 years, many people, many child will be died. So one got it, made it open. No need to patent. Sometimes patent is good, but it's better humanized. So now we got the point little bit how from the popular idea, traditional knowledge is goes to the experiment, perfection, both in traditional knowledge, rice water, and salt, is art. What is it? Is the science. In water, everything is specific. You have to need 1 liter or half liter water, and everything is constant. But in the extra water, it is nothing constant. Okay, this is fluctuation. Okay, but IT is there. IT is there. This is the idea. And this is the polishing instance. You have just polish it. Okay, so drug development, more or less same process. Anti-diarrheal drug. So during diarrhea, we lose a lot of water, right? Lot of water and lot of salt. My question is, why we're taking, instead of taking salty water, why we're taking, why we are adding sugar? Anyone know it? Anything?
Consumption.
He's right. So this is channel. Sodium transport and glucose transport are coupled. Coupled means sodium cannot go alone. So it glucose. If glucose enter, sodium enter. It takes many years. So what is working for diarrhea? How? What is the mechanism? It is not known. Long time. Someone discovered it. He got Nobel Prize. Now, came to our main topics. Okay, the development or discovery of drug. So, plant materials is the best source, best natural sources for the discovery of drug. Initially, 100% drug from plant sources. Nowadays, 50% of the drug, all the drug, either directly or indirectly, still there is no replacement, still in plant sources. And from plant, the plant source, the maximum is that it's a chemical agent. It is a molecule, simple molecule. We call it chemical agent. So it's a hard work. How from a plant materials you can get a single one. So you are sitting in a chair, the chair, best wood. The wood came from the tree, mixture of the tree or the unused tree. You take only the good part and you make your furniture. Same is true for the plant materials. So after plant materials, collecting the plant materials, okay, if you make extract, then different column chromatography, different separation, different extraction procedure, you have to isolate it many compounds. Yet single plant contains thousands of compounds. And thousands of compound has 1,000 kind of activity. But in plant, normally you are taking some edible things for diabetes treatment, where people take it Kerala, right? So edible part, it is safe. It has some chemical that has good property and some chemical has bad property. It minimizes it. But once you purify the compound, it may good, it may bad. Therefore, if you even for the known plant materials you are taking everyday life as a food, herbs, but single compound may toxic. Therefore, this single compound identified, it goes through some process, either it is safe, secure, effective, many things. Okay, this is the drug discovery start. So the active ingredients, for natural products, isolated and purified in chemical methods. There are many different chemical methods. Okay, chromatography is there, solvent extraction is there. Many ways you have to fraction it. Okay, so students, how many you are unmarried? Can you raise your hand? Unmarried. How many students are unmarried? Mashallah, mashallah. Okay, okay. So you are unmarried today, tomorrow, or day after tomorrow, 1 month later, 2 months later, 1 year later, you got married, right? First, you have to declare, yes, I'll get married. Then what will father and mother do? He'll make you just make all the relatives, they circulate. My son or my daughter get married, please ask. Photographs, please send photo. So from Facebook or different ways, you are getting thousands of photographs. Okay? Out of 1,000 photographs, first see the photograph. Out of 1,000, okay, you select 100. After selecting 100, okay, matchmaker, then your father, your mother, your cousin sit together. Out of 100, 90, no need. You see photograph, first this photograph. Okay, seems good. Biodata here, academic background, this, okay. Photograph 2. Photograph तो सही है, academic background, location match नहीं कर रहा है, discard. ठीक है ना? In this way, out of 100, you are getting 10. Then उसको पता करो. खानदान कैसे है? ठीक है ना? So अभी की what is this father, mother, मामू खालू है ना? Right? Then 5. Face to face discussion. ठीक है ना? Then After 1, 2, 3, 4, 5, came to home and you have to do some statistical analysis. Eh? इसका good side, bad side, good side, bad side, good side, bad side. Out of 3, 2. Now you have 3. This is very, very critical time, decision. You have to take many things. You need FD approve. FDA means father के approval चाहिए, auntie के approval चाहिए, और किसके approval चाहिए. So finally you got it, right?
Yes.
Sorry. Anyway, age ठीक है ना? You got married, happy life. I mean, but after 1 year, what happened suddenly? Sometimes, है ना? So drug discovery process exactly same. Drug discovery process similar. This one before marriage, after marriage. You got my point, madam? Sometimes to get some chemistry to make it excitement, refreshment. Take it. After this, you got my point? Okay. I'm not professor, but I'm very active, dynamic. Okay. Ajba told me to sit there and no. So if you have any question, okay, curiosity, ask me.
So why it is named as drug discovery and development? From the past 100 years, it should be named like medicine technology because we are using it for therapeutic purpose.
So, as I told you, initially, the medicine came 100 years back. Drug came 2,500 years back. People use it, huh? People use it. So now it is the city of the name of the city is Islamabad. Previously it was name was Pindi. Okay. So now Faisalabad is name. Initially it was so it changed. Sometimes change is good, but because sometimes many miss the word farm. So once it started, even the function is differentiated, didn't change it. Because initially drug was developed initially. Medicine came because the practice. These people use 2,000 year practice. It's very difficult to replace this term with this term with medicine. You got my point?
Yes.
So one is purification of chemical agent from plant sources, different procedure. We call it pure compound. Another is if you got the compound, if you make it in laboratory, someone is organic chemist is there, so you can make it synthesized by laboratory. Okay, but so one is natural product, another is the natural compound, another is the synthetic compound. Which one is better? No, for medicine, synthetic product is better because natural products, as I told you, thousands compound is there. From thousands, different procedure, you got only one. Chance of their contamination is there. Okay, contamination is, it is not 100% pure, maybe 99.99% pure. Some impurity may be there. Okay, some instability may be there, but those are chemical synthesis chemical, they're more safe, better than the natural compound. So synthesized compound can be obtained from natural sources such as they are totally synthesized. Chemical synthesis products generate greater purity and at low costs, and they have synthetic drugs at great advantage. Reduce side effect. Okay, it save you time, save your money. Okay, you need small spaces, small laboratories enough. For chemical synthesis, you need a good big laboratory. So drug discovery requires a knowledge of biochemical process involved the metabolism of the body. So the disease all the disease in human body, it is a disturbance or disorder of metabolism. Either your someone enzyme is upregulated or some is a downregulated. If it is upregulated, make a compound so that it can downregulate. If it is downregulated, make agent or compound so that it can upregulate. This is the drug. Okay, this is the drug. Yes, drug discovery, it's multifactorial process involving extensive screening, testing, optimization, lead compounds, potential new drugs, and therapeutic use. One is drug, another is the phases. A chemical substance of known structure other than the nutrient and essential dietary ingredient used for the biological living organism. Discovery phases. One is discovery phases, another is the development phases. See it here, photograph of cycle, drug discovery cycle. So compound, lots of compounds here. So you are using in vitro tested primary assays. In vitro tested means either it is first we have to check either toxic or not toxic. Okay, then we got it secondary assays, toxicity, metabolism. Okay, then it can goes clinical candidate. Option 1. Option 2, this one it can goes, if you got the chemical synthesis, we can modify the compound with the structure activity relationship. Okay, then this one. Another thing is we can design, we can synthesis, and we go in vitro test. Okay, there is no straightforward pattern. There is, we call it crosstalk, cross-linked, many ways. Like plan A, plan B, plan C. If plan A fail, sometimes we can do, if we do plan A, plan B, plan C together. So if you want 3 pathways, at least chances there, one pathway may work. Why new drug development is needed? As I told in my lectures, patients are different. Paracetamol, you're taking paracetamol, right? Normally, when dose was developed, normally per kilogram body weight. It was designed during discovery, this per kilogram body weight. But in reality, it is very impossible to make personalized medicine. So 500 milligram paracetamol. So you're taking it, your BMI is 20, someone BMI is 15, someone BMI is 30, right? So if your BMI is 15 and someone BMI has 30, your metabolism, his metabolism is different. Okay. So patients are different. Medicine is the same. It may or may not work to all. As I told, one size fit all. It's very uncommon. Only few countries, very few countries manage it. Very few countries. One example is Japan. Okay. All the female, same shape. All the houses, you cannot make house your own ways. In every city, there is a design A, B, C, D. You have to make it. You cannot make your own design. So you can find in the city, all the buildings are the same. It makes your life. So what they do, if all the building design are the same, then just under construction, after construction, all that is one, everything is ready-made. By overnight, they can make the building. Without the time is money. So one size doesn't fit all. If you put the market, some shopping, you can put the salwar kameez. One is a small size, one is medium size, one is L size, XL, double XL, different size. If clothes has different size, you're taking food. So you have 5 brothers and sisters. 5 brothers and sisters, you have 5 different kind of liking and disliking. Someone like chicken, someone like fish, someone like vegetables. So even same family member, 5 family members have 5 different choices. 2 brothers and sisters, twin brothers and sisters, 2 brothers and sisters, two different subjects. Someone is studying biochemistry, someone was in chemistry. So if it is for diversity, if the same medicine, it work to all, may or may not. See, I'm giving example of diabetes. On left side is the diabetic patients, right side is also diabetic patients. So left side patient, okay, this is a beautiful lady, 14 or 15 years old, she's obese, she's diabetic. Take it now. She's diabetic. But right side, you know who is that? Okay, he's from Pakistan. Two different people, one is Western, another is Asian. Two different people, their diabetes, two different types. Left side, Western people, she has insulin resistance. Okay. Right side, insulin secretion deficiency. He needs insulin, he don't need insulin. Pathology is different. Sit here. So in developed countries and developing countries. In developed countries, you sit here. In developing countries, normally their diabetes start, maximum diabetes start after 65 plus. Okay, 65 plus. That means after age of retirement. But in developing countries, diabetes start at middle age, 40 plus. That means after 65, you are becoming grandfather or grandmother, right? But at 45, you are working, you are active, dynamic, productive, and reproductive. So, a reproductive age pathophysiology and retired age pathophysiology is different. Another good example. So, one patient, the left side, he's from UK, and right side, he's from India, Asia. Say, both have body weight, BMI, same. We call it, they call it OIY syndrome. It's got a name here. First name start with Y. Right side, first name Y start with Y. We would call OIY syndrome. Two peoples, BMI is the same, two different regions, but their body fat is different. Okay, someone has central obesity, Asian people, and European people have whole body obesity. It's got fat distribution in whole body. But in Asian people, in central obesity. So even same BMI, but fat distribution is different. So students, you are getting in intermediate or O level. So you get result, it's GPA or mark system. GPA. So you are getting GPA 5, someone getting GPA 5. Someone get 90% in mathematics and English 60%, 70%. Someone get English 90% and math 70%. Both of you got getting GPA 5, but both are same. Someone has interest on English, someone has interest on mathematics. Given the result is same, but their interest is different. Pathology is different. The phenomenon liking is different. Same is true. Passion is different. So I give another example. This is the WHO criteria. See it here. The Asian people, it is less than 18. See it here, normal weight. 18 in Asian countries, 17 to 22. In overweight, it is 23. But in other Asian countries, non-Asian countries, 25. Obese, 28. In non-Asian countries, 30. If criteria, you make criteria for the Asian people, BMI is different. Okay, or cancer is different, food is different, everything is different. So this is the time, this is the thing, this is the May, this is May. Automatically is different for Western population. So number one. Number two, the DNA sequence similarity. So human to chimpanzee, 98% similarity, right? and human to one. So one human to another human, two different human. What is the DNA sequence similarity? Anyone? Be two different people, right? Me and you. DNA sequence similarity. What is the percentage of DNA sequence similarity? Anyone? 99.99. Okay, that means only 0.01 is difference. So it is 0.01 difference in 100. Multiply 1,000, 10,000, 10 million, 10 billion, 10 trillions. In trillion, this pair changes many. Okay, it called a small changes make a big changes. Difference only 0.01%. See it here. A drug that treats and save one person might harm another due to SMPs. Okay, SMPs. So one drug, we call it, what is the name of this? Resolene. It normally is used for diabetic patients. It improves the insulin resistance. This drug causes death more than 30 patients. This is a study. That means one medicine cannot save, one medicine save your life. Same medicine can harm others. So a drug that treats, save one person, might harm another. So Professor Atar Rahman, you know, Professor Atar Rahman is the first chairman of ACC, and he was former coordinator general comstech. Okay, he's the first, become the first Pakistani for the genome mapping. His genome sequence was done at ICCBS. Okay, nowadays we are giving the medicine, are giving the prescription. Please this one. Maybe after 10 or 20 or 50 years later, you have to go there. Okay? You have to go there, and this kind of sheets will be available for you. Your DNA sequence. First, check it. What kind of DNA sequence is there? Okay, time is coming. Okay, so make it own medicine. Maybe this, you got my point. So SMPs, single nucleotide polymorphism. This change one people to another people. So because population to population variation as well as regime to regime variation. Therefore, so one drug, one is our genetics is different. Okay, and normally our everything is depends on the package we call it epigenetics, environment, lifestyle, many things related. So only one medicine cannot save all. Number two, during the COVID vaccine, those pharmaceutical develop. So those pharmaceutical develop medicine, they want to sell it. Everybody want to take this one, pre-ordered. Even you get money, you will not entitled to get vaccine because those are powerful. Those are powerful. First, pharmaceutical company have to give to them. The COVID gave us lesson. That means even if you have money, you can purchase, you can buy. Even if you want to purchase, company will not give you. You cannot do it. First, they will give Canada, Australia, USA, then. Okay. Then, therefore, we have to think, we have to build our own design, own drug. Okay. For discovery of drug, designing of drug, development of drug, there is no prediction. Okay, like getting married. So after 10,000 biodata, you got married, you think it will work. She will do well, everything will take care of all things. But it may or may not. In those who have knowledge, don't predict. Those who don't predict, no knowledge. For drug development, there is no prediction. Okay, so therefore, we have to do our own research. We have to think for our own drug. Okay, With this, we'll have a little break for refreshment, then we'll again start, inshallah. Please.
Right, the examples explaining how the drug can be different and very different. And I will ask the audience, any questions of the first lecture, they can ask Dr. I will just add, when you talked about glucose, if you see the property of the glucose as a the glucose also has anti-inflammatory and antibiotic activity.
Pena. Okay. Yes, my dear brothers and sisters, you take tea and coffee. Now you are energetic.
Yes, sir.
Tea and coffee contains caffeine. It's a drug or medicine? Anyway, thank you. So one is prediction, another is prediction is always prediction. It may or may not. It may be 0%, maybe 100%, 10%, 20%. Either it is 0 or 0. Now we are coming to the most important topics. Drug development, the process of bringing in new pharmaceutical drugs from marketplace. Okay, marketplace and lead compound has been identified through the process of drug discovery. Quinine, no, it's malarial drug. Okay, it's also a study of folklore society. In African people, they're suffering from malaria. So normally in Bangladesh, Pakistan, this kind of world, there is some we call dengue. It's due to mosquito. Okay, same is true. Malaria is also a mosquito disease transmitted by mosquito. How normally for dengue, to prevent dengue, there's no mosquito in my room. What we do? We use coil. Same is true. The people in Africa, they use some tree buckle, make it firing, and they make some coil, not coil, it is some buckle, this wood, they make some smoke, they use the smokes. These smokes prevent coming mosquito in their house. So this is their idea. This is the art. And scientists, what do people think? If this kind of smoking prevent the mosquitoes, maybe something is there that can prevent malaria. From this active ingredient of this plant, it's a medicine. They develop it as a quinine, the malaria. And Paul Atlas, he's called the father of modern drug discovery. He discovered many drugs, including morphine, cocaine, and aspirin. Target validation, lead identification, and lead optimization. All the process we'll see little by little. Sit here, I make a draw. Okay, one is disease identification, target identification, target validation, lead generation, lead profiling, lead optimization. This thing is called drug discovery. Later it goes to the development process. Initial stage in the animal model or animal. Okay. Then from animal to human, it is called a development process. So before human, it was the discovery process. Drug development is a risky, expensive, and lengthy procedure. Take number, and it takes 8 to 20 years. And for biotechnology, pharma, it takes 7.7 according to 1994. 7 billion for the biotech pharmaceutical, biopharmaceutical. And pharmaceutical, it's almost $15 billion. So the pharmaceutical company in Bangladesh, Pakistan, third world country, we call it normally packaging company. Mostly, honestly speaking, no, you know. So in Bangladesh, Pakistan, we have garments factory, right? Garments factory, garments industry. Okay, it is garment industry, but really think what are we doing? We are purchasing the clothes, machineries, everything from outside. We are doing only stitching part due to low cost of labor. Same is true in third world countries. Pharmaceutical company really, in true sense, they're not pharmaceutical company. Okay, we purchase formulation. Okay, and we do only the formulation. We purchase it and we make it drug or medicine and sell it. Okay, but really, once medicine came from the conception to the market, it takes many years. Many years. Yeah. Same is true. Eh? When we start your education, class, playgroup, nursery, 1, 2, 3, 4, 5, 6, 7, metric, inter, honors, masters, then you are getting married, right? So it takes 18, 20 years to prepare yourself to be a good candidate in practical life. And that is office at home. Same is true. It takes time. And everything needs times. There is no shortcut. No shortcut. See here, normally, so one chemical or compound you got from some sources, plant sources, fungus, and so on. Then one is discovery phases. Another is development phases. The discovery phases, we do a lot of testing. So it makes sense to for the discovery, a lot of things we do for the safety, efficacy for the development phases. Okay, preclinical testing. Preclinical testing, it is done normally in animal. Okay, so drug efficacy, optimal doses, potential side effect, and toxicity. These are the things we have to do before going to the human. See it here. So as I told you, getting married, similar story, 10,000 compound. Then you tested 10,000 compound in vitro, in test tube. Then maybe you got 25 compound is 10,000 compound testing for either some activity, maybe one activity, one or two activity, either is active or inactive. You got 250 compound, they are active. Okay, these 250 compound you did preclinical study in mouse, cat, guinea pig, rabbit, right? And you find out of 250, only 5 compound in clinical trial. This means preclinical is done in animal. Clinical, it is done in human. So once in vitro, 10,000 to 200. So when you transfer one formation to another formation, test tube to animal, not all works. So 250 clinical is working in vitro, it is active. They have some activity. But in clinical trials, Out of 10,000, only 5, maybe 5, maybe 1, maybe 0. That means in vitro test tube. In test tube, we are considering only few things. So temperature, room temperature is 25 degree, or we make the reaction in 37 degree, right? We make the pH 7.4 or 7, maybe. How many typical? Maybe we consider pH, temperature, or 2 or 3 things. But in human body, in the cell, hundreds, thousands, million factors. So in the test tube and in the tissue is different. Sometimes in online, you ordered something, you are getting different. This photography is very beautiful, but when we got it, it is not same. Okay, same is true. So the test tube and human. In test tube, we are considering only 2, 3, 4, 5 factors. But in cell, it is thousands of factors. It is living, another is non-living. So it is pub compound in FDA. If you submit FDA, the FDA lots of documentation is needed. So finally, you get approved one. Okay. So after preclinical study here in the animal, okay, we do clinical trial in human. We call it phase 1, phase 2, phase 3. Then it came to the market. You see a little bit here. This is a good clear picture here. So this is basic research, early discovery, preclinical, right? This thing is a laboratory in vitro. Preclinical, it is in animal. Then it comes clinical development. Phase 1, phase 2, phase 3 clinical trial. Then FDA. After FDA approval, it will market. So after FDA marketing, may it consist or may not. Okay. So see it here, 100,000 compound. Finally, you get only one compound. One is here, human safety, human efficacy in the phase 1, phase 2, phase 3. We check it, human efficacy, either it is most important is a drug, even if it's highly effective, high has efficacy. So it must be non-toxic. It must be non-toxic. To be a drug, it should be non-toxic. So what we are doing? We are doing some basic research. Then we get some translation research. Then basic research we are doing in the lab. Sometimes give it in the animal, we call translation research. And the translation research to the clinical research. So step 1, step 2, step 3. So as I told you, so thousands, some hundreds of compounded testing, 10,000 compounded testing. Out of 10,000, you are getting 100 compound. This we call candidate compound, lead compound, have activity. Then lead compound, out of lead, okay. So those out of 100 compound, maybe clinical trial 5 compound. One is lead. This 5 compound will be called hit compound. Take it up. So these 5, one is many compounds, then active compound. Those active compound we call it active compound, we call it candidate compound. With this candidate compound, some has poor activity, some has moderate activity, some has strong activity. Those strong activity we call it potential compound, or lead chemical, lead component, lead molecule. Clear? This lead molecule, those are from this lead molecule, we have to hit. Which one he will hit? Market or the drug? So then, this is the study. Then the study first begin in the test tube. As I told you, either it has activity or no activity. If those have no activity, no need. For those have activity, first we have to check their toxicity, whether it is toxic or non-toxic. The in vitro, this means the test in glass, outside the body, test tube. So initially, people do in the test tube, skinning the test tube. It takes lot of energy, lot of chemicals. Nowadays, we call skinning in microplate. 96 cell plate, 96 cell plate, and that is 384 plate, another is 1536. That means the plate, initially the hole is big. Now we make it 96 well, now it made a whole size. Initially it was 1. You have to test to use 1 ml chemical, this. Now 96 cell plate, 100 microliter, 200 microliter. Uska the depth है ना 300, total 300 microliter. So now it makes 100 microliter. Now it makes 20 microliter. So in this way, a small space, you can test thousands of compounds, single go. You just add the compound, different oil, right? The other chemicals you have to give by machine within few seconds. Only different compound. In this way, thousands and thousands compound you can check very short period of time. Okay. So see here, this is the basic research. Okay, and here is the clinical trial. In between, clinical trial. Lab is here. Lab is in the university. Patient in the hospital. From laboratory to hospital, who move here and there? Chua. Anyway. They make a breeze between you. So these are a few examples of in vitro bioassays. Sometimes we check DPPase assay, and sometimes DNA level assay, RNA level assay, and most important, integration assay. Now, what kind of drug you want to do it? Depending on it, some 1 or 2 tests we have to do. Sometimes common test is do either it is antioxidant or not. Okay, antioxidant, some are common test. Then, depending on their, if you work diabetes, either it is activity or not. If you work for cancer research, either it has anti-cancer activity or not. Depending on your target, you have to fix your assay and pass this on. One thing is common is the toxicity assay. Either this compound is toxic or not toxic. This is common for all. This is common for all. Getting other to give an example, getting married. So everybody should. Wife है ना, इसका common criteria क्या होना चाहिए? रोटी पकाना. Common है कि नहीं है? Almost common. Something is common, something is very specific. Another is after chemical in vitro test, then we'll do in vivo. In vivo, sometimes we call it sometimes in vivo. It is in living system. Living system. So living system, we can do it in animal, or we can do it sometimes in organs of the animal. In animal, we call it acute toxicity, chronic toxicity, then clinical trial. This kind of thing in the living condition. Example of in vitro, in vivo assay, many. Okay, many. So depending on this one, anti-cancer activity, drug metabolism, anti-parasitic, anti-obesity. This kind of activity we do in vivo system, living system. So this preclinical study, one is in vitro, another is in vivo. As I told you, in vitro it is in the test tube. In vivo it is in sometimes in vitro. Remember, in vitro, there is a T, right? T for test tube. In vivo, sometimes your mobile, sometimes you make it in vibration mode, right? Sometimes you make message called sometimes if you have live, you have sign vibration, heartbeat here. So don't confuse it. So sometimes we teach the students, even make mistake. In vitro, T for test tube. If T for test tube, D for light. Clear? Normally, in preclinical study, we do in I think it gives for in. Mouse. Okay, we do it here. So what we do normally if we give, so as I told you, it easiness and diseasiness. So if you normally, so if easiness is attained, it always straight. Diseasiness, what happens? First thing, if you give anything to the rat, uska tail hai na, that means it don't feel comfort with this drug. First, if you give the medicine, taking medicine, mithai hai na, you take some juice, this one, it is good juice, good impression. But juice, but it's not good impression, first it shows your appearance. Same is true. If you do any test in the rat, it's behavior, physical appearance. Okay. So sometimes physical appearance, you have to see it first. Because toxic effect later. First physical activity. So this kind of we do efficacy, toxicity, and pharmacokinetics. So all the things, hopefully. So one of my students, Abhida, she's working on it. So she's very good in animal handling. Okay, extraordinarily good. So if for animal experimentation, she knows better than me because she's a real player. Okay. If you want to do some animal experimentation, so consult with her. She can give more practical approach. So how to handle those are married, है ना? Someone has child. एक ही है, चुहा नहीं है, मेरे बच्चे हैं. You have to take love and affection. You have love and affection to the before doing any experiment for animal, you have to understand the animal behavior. Okay, बच्चे हैं. ये जिंदा है कि तुम्हारे डिग्री मिलेगा. इसको make him happy. The good food, good mood. Change their bedding. First understand. So first understand its behavior. The bedding, good bedding. So during animal handling, I will teach it. Preclinical study. First, most important is the toxicity study, right? Toxicity study and drug interaction. And once the experiment is done in the test tube, in vitro, preclinical, in that, then we'll move to human. So before going to human, I want to know attention to the pharmacy background, you know better. To the non-pharmacy background, there's a term we call placebo. So everybody knows this term? Okay. Those don't know, everybody knows it well and good. One is capsule, both, one is green, another is red. Both are capsule. Okay, one is green, another is red. But inside the ingredients are the same. Only difference is in placebo, the active molecule is not there. In medicine, active material is there. So it is written here placebo, but in case of real test, nothing is written. Nothing is written. One is green, another is red. I am giving, I know Green is the placebo, red one is the drug. I'm giving the patient ABCD this one. Number one, I'll give green. Number two, I'll give red. Number three, green. Number four, or vice versa. Right? I know which one 1, 3, 5, this placebo. 2, 4, 6, it is the drug. I know it, but the patient, he or she know, she doesn't. That means out of two, patient and me, only I know, she don't know. That means out of two, one is known, another is, it's called single blind. Patient, he don't know. Taking medicine, acha lag raha hai, feeling, sometimes placebo effect. The drug, placebo effect is better than the drug. Psychology. डॉक्टर ऐसा होता कुछ नहीं हुआ आराम करो बस 1 महीने बाद आराम करो खाओ ये खाना पीना खाओ आराम करो एक करो ठीक है if so sometimes a drug comes from somewhere red and green even I don't know inside I am giving that means okay red and green please write red and green I am giving red someone green someone getting result. Okay, the patient don't know who's on drug, who is placebo. And I am also don't know who's on drug, who's on placebo. It's called double blind. Got idea, clear idea? One is single blind, another is double blind.
Double blind, only company knows who is going to provide you.
No, company don't do this. We call agent. Some agent is if company knows it, it will be very dangerous. Somehow will make result in your favor. Somehow we call conflict of interest. Someone they can make a result. Someone make a connection. Who is doing this test? This patient किसके पास जा रहा है? Medical representative ना MR है ना? उसके पैसे लगा दो. Green किसके पास है? Red किसके पास है? पता करो. It will make they make the result in their in favor of them. People don't know it. So animal model disease stress we will discuss later. So as I told you, first behavioral study. Either it behave well or not disease. If behave well, that means it medicine or drug or active compound, lead compound may work. Ex vivo. One is in vitro, another is in vivo, another is we call ex vivo. Anyone have ideas what is the in ex vivo?
Tissue culture.
No, not tissue culture. Tissue culture, cell culture, it is in vitro. So normally sometimes people, so you can collect some heart or some eyes from the dead body, right? You can take it, eyes, and you can put in your someone eyes. It active or inactive? Active for few hours. You have to collect the organs. Within 1 hour, 2 hours, this one. I have to do all the procedure within 6 or 12 hours. That means even human life is not there, but organ is alive, active. It is you come, you take the eyes from someone and put it someone. In between, it's outside the body. Outside it ex vivo means ex means exit. It is outside the body. So if you do some experiment, so in the rat we do normally pancreas. If we take the pancreas or any organs, and if you do some experiment within 1 or 2 hours, it is called ex vivo. It is in between in vitro and in vivo, and it is more physiological than in vitro. This is the advantage. In vitro, ex vivo, now we call it in vivo. In vivo, we call it the clinical trials. So in the clinical trials, there's two kinds of study. One is we call observational study, another called is interventional study. All the clinical trials, it is interventional study. So one is observational study is, so you are studying student here, 30 students, we are giving tea, tea and coffee, free of choices. Someone taking tea, someone taking coffee. We just observe it. What is the effect? We have a concerning. This is called observational study. Okay, and if we give some medicine, this, we call interventional study. We have to intervene. That is, so this is, I am giving one example of my own research. So what I am doing, I am working for last many years for diabetes. So people India, Bangladesh, Pakistan are very popular of tea. We always, in between there is a tea break. So what are the things is reduce the blood glucose. I did some animal experimentation as well as some human trial. Okay, I make tea bag and give patients. What is the taste, flavor, whether it is reduce the blood glucose or not. Okay, me and my group working on it. Okay, because for the marketing, there is issue, we need clinical trial. So recently I submit a project to the Ministry of Health. What happens? So what we'll do, we get, say, how many patients? 60 patients. 60%, 30, right? These 30% will give normal tea. This 30% will give my tea. Okay, TA, TB. 90 days will take the tea. Okay, then it will call, we want to see the effect of my tea. It's called interventional study. It's clinical trial. Clinical trial. Okay, clinical trial. It's called single blind clinical trial. I know it, the patient don't know it. Okay, so A, B, 90 days. Okay, then after 2 months, we change it. A will be B, B will be A. It's called cross-sectional study. Got it? A will be B, B will be A. In this way, that means same patient, they will initially they took normal tea, and after 3 months, they will take and diabetic tea. So it is single blind and it is cross-sectional. Beauty of cross-sectional study is the patient itself control. No need volunteer control. Option 1. Option 2, A, B. A is normal tea, B is anti-diabetic tea. So after tea, we make the patient here. We change the tea. The group A, diabetic tea, group B, normal tea. Option 1, we can cross the patient. Option 2, we can cross the medicine. This is the beauty of cross-sectional studies, no need control. The patient, sometimes either initially he was controlled, later was patient, or initially he was patient, later he was controlled. But in between, there should be some what we call washout part. So this is a good example for those people who are working here, sitting my workshop. So any people you are using many plant materials, this one plant materials, but even sometimes your folklore society, folklore knowledge, you know, this one is good, this one is good, this one good. You can do by your own way. You can do a small number. You can start with feeling good taste, with good activity. You can take by yourself. Medicine cannot help you, but this kind of homemade remedy. So initially, my father, my mother, my daughter, so sometimes they never ever going to the hospital. Initially, if there is something, they took homemade remedy. Even so in Pakistan, Bangladesh, even your child is sick, you go to the hospital, you are getting medicine. But USA, doctor don't give medicine. Initially, flu, this, no, they only give honey. If after 5 days, 15 days, 10 days, 15 days, this is not recovered, then they think for medicine. Western country, they are developed country, but medicine, all medicine are poison. This kind of cross-sectional study you can do by yourself. Easy. So what about around you? If it is because the tea we make it is all are edible, no risk. Sometimes in the tea we add cinnamon, darchini. Sometimes we bay leaves, tejpata. It gives some flavor. Okay, and the bay leaves has very good antioxidant activity. So if you boil it few times, the water color will be little bit yellowish. If you add tea back there, you see, without bay leaves and with bay leaves, there is difference. Once, one professor from US came in ICCBS, he has some throat problems. Okay, he couldn't talk it. Okay, so I asked my students, please do in this way. And my formula, the student add adrok, ginger, additional experiment. And believe me, the professor take the tea in the morning 2 times, but evening 100% time. Okay, so tea. Okay? Normally, the people in Pakistan, Bangladesh, if you have good mood, let's have a cup of tea. Sad mood, tea can solve many problems. You can, a good mood. So this tea, you can what you want tea. You can take the tea as well as you can take as a medicine. For antioxidant is the all medicine for all. Now, the clinical trials. We start from the clinical trials. So, in clinical trials, there is lots of regulatory approval required, as well as your initial approval is needed, individual versus collective benefit, 100% volunteers, informed consents. So, informed consent means you have to give the medicine to patients, right? Yes, no? No. Initially, first we have to do clinical phase 1. It is in volunteer, not patients. Healthy subject. In healthy subject, we have to give our drug, either it is, they can tolerate it or not, toxic it is not. And most is verbal and written information. Normally, I am working on it. The patient, we have to ask the patients. We have to explain the situation. We are developing this tea contains these. We are making it for safe side. What I do, make the first cup tea, I take it by myself. Okay, then explain it. Then that people normally they are very much afraid. Okay, very much afraid. We make tea, then I ask, you can take anyone. If I take cup A and give you cup B, they have confusion. If you free choice, in this people are not willing to join in the try. Therefore, sometimes what happens? We ask people, okay, so now I have lot of work, this. Okay, whole day work, you will get 2,000 rupees. We'll pay for 2,000 rupees for you. In this way, we hire the people. By paying money, we take it. And we have to explain verbally. And nothing will be wrong. If some adverse effect, we'll take care of the responsibility. And sometimes, then after explaining everything in verbal, we take written document and sign it. This because for further step, all the documents, his name, identification, NID, all the things. Okay. Normally we make it, we never disclose it anywhere, only for research purposes. Other than okay. Even sometimes no one willing to share his mobile number, WhatsApp number, so on, or NID number. Yes.
Sir, what is the criteria for selecting the subject for clinical trial? Like some drugs don't have any toxicity, but they may cause allergy like penicillin. Some people are allergic to penicillin, but we have 0.1% difference in our genome. So it may cause allergy or it may cause any other serious consequences that may cause it. So what should be the criteria for selecting the subject?
Only one criteria. Only one, healthy volunteers. So either normally we check it, we asked it, okay, either BMI within range or not. We asked, did you take any kind of medication, this. So those are no medication. No medication. It appears healthy, seems healthy. At genetic level, it's impossible to understand. People don't know their blood group. How can I expect they know their genetic sequence? At this moment, only one criteria.
2,000 rupees. Oh, you will not tell you that whether he is or not. There is something wrong.
Okay. He told good things. Sometimes there is study, customs, custom officer, they work. So you have to give some money. In Bengali, there is a proverb, no, he's very honest, he don't take me. Koto khaina. Means how much money he don't, his rate is 50,000. Usko laga do. ठीक है ना? Sweet 2,000 not working. Double कर दो. Anyway, okay, in clinical trials, there is a different partners. Okay, normally this clinical trial is done by not company, not hospital, we call it CRO, Clinical Research Organization. They work, sometimes they are like their contractor. Okay, their contractor, they got contract from you and do their work. In the road, your government giving some money for the making a good road. The contractor will use it, may or may not. But people will use it. He's just doing on for the third party. Okay. Same is the CRO, it's third party. Not belong to patients, not belong to the company. CRO. It means partner on sponsor, CRO, in the investigative team, ethical committee, regulatory committee, and study subject. Many partners for their contributor for their clinical trial. Patients, CRO, then investigative team, pharmaceutical company. Normally, pharmaceutical company give the money to the CRO. Then this one, in this way, it's not only single component, many contributor. Yes.
What are the requirements for ethical committee?
Ethical committee, but all the institution in RTCBS or I am working in Dhaka, we have an ethical committee headed by doctor. Okay.
It is mandatory to be headed by doctor.
Yes, it's in Bangladesh. I don't know here. In Bangladesh, in clinical committee, it's headed, there's a committee in national committees there, as well as everywhere there is an institutional committee. If you work on human subject, I am working in T, so be headed by doctor. I have to submit the project proposal first. What is materials or methods? Okay, then sometimes they took us interview. What you want to do it? Then considering everything, they approve it. So this clinical, we call ethical, either it is ethical or not ethical. So in animal housing, so I was a member of ethical committee in ICCBS. So I am working on animal, is a procedure. We will make a blood glucose test in rat 4 times, 0 minute, 30 minutes, 1 hour, and 2 hours. What he or she will do at 30 minutes, he'll cut the tail, cut the blood. After 30 minutes, cut the tail once more. Okay? If it is tail here, cut it here. 30 minutes, cut it up. After 1 hour here, after 2 hours here, I repeat the project. No. You want to jinda chahiye. 1, 2, 3, 4. No, you can collect blood from vein or somewhere. Udar cutting. So if you cut 4 times, the animal will be easy or not easy?
Easy.
Not easy. So you get the result. Okay, it is not true reflection. Either false positive or false negative. I reject the project. Similarly, there normally either ethical or not, so it is animal. I reject the project. It's by human. Either it goes anything against with him or her. If 0% something goes against him or her, modify it. So getting clearance from ethical clearance, it very, very difficult in Western countries. But here, it is a bit easier. Therefore, many big companies, they do their clinical trials here.
Yeah.
Okay, because very difficult to get ethical clearance there. Okay, it's a good opportunity for us to make CRO. Okay, now clinical trials. We call it human trial. Sometimes call it clinical trial. Phase 1, phase 2, phase 3, phase 4, but there is a 0 phase. So normally, the 0 phase. So anyone can tell his or her birthday. Say January 2, X, ठीक है, 2020. Then January 2, 2021. His age is, what is the age? 1 year. The first birthday, right? You agree? Normal people do. So after 1 year, people make it first birthday, right? But it birth start 10 months before. After 365 days, you are in the ark. But 0 days, you are in the inside your womb. That means if you really think it's first year birthday, it's okay. But you're in social aspect, your age is 2 years. But biological aspect, your age is 2 years. 1 year inside your mother, 1 is out first birthday. Okay? That means 0 day. Same is true in phase clinical trial, phase 1, phase 2, phase 3, phase 4, as well as phase 0. So clinical trial, this is the most time consuming and most expensive. This is the period. First, see the phase 1. Phase 1, it is done on healthy volunteers. Healthy volunteers to check either the drug has any toxic effect or not. Simple question, simple answer. Number 2, the doses. What is the maximum doses it can tolerate? These two things mostly, these two things, 2 points that focus for the clinical trial. Okay? Normally 50 to 100 volunteers, human toxicity, tolerated doses, 70% drug candidate pass. It is in normal subjects, healthy subject, normally 70% candidate drug pass in this phase 1. Now came to phase 2. Phase 2, initially phase 1, it is in healthy, but phase 2, it is in patient. Say 300 patients. Then we call it single blind. Patient don't know, but who is giving this medicine? Single blind. We check it either safety everywhere. Every toxic effect, phase 1, phase 2, phase 3, applicable everywhere. Toxicity is the common. But here we check it safety and efficacy. Either it is safe or not, either effective or not in patient, in a specific disease. These are the 2 things that focus on here. Okay, in phase 2, either it is safety and efficacy. Either it is giving the drug, it is working or not working. If it is working, it is efficacy is 0%, 10%, 50%, or 100%. Right? Now come to the phase 3. In phase 3, normally what we do here, most expensive and time consuming. Normally here phase 2, say 300 patients. Okay, but phase 3, 1,000 patients. The big difference here. So because one is phase 2, phase 3. My question is, why we already know the safety, efficacy of a target compound in patients? My question is, why we need phase 3 clinical trial?
प्री क्लिनिकल ट्रायल में सर हमारे पास 15 की तादाद भी ज़्यादा हो जाती है और साथ में अदर कंडीशन तो भी फैक्टर्स मजीद ऐड हो जाते हैं.
Okay. Any other?
There is a DNA difference is 0.01%, so more patients will included to check whether there is some toxicity.
Anyone?
Comparison with the other drugs.
Anyone?
More biological parameters.
Anyone? Look, one thing is big difference is phase 2 and phase 3. Phase 2, it may be single center. All the patients from flower, all the patients in city. But the phase 3, it normally should be multi-center. Multi-center, it may be multi-country. So one drug, if you test one drug in Karachi people, or one same drug, Dhaka, no, in Punjab or KPK, same drug may not be the same to the Karachi and KPK patient. Right? So the phase 3, so once you get the idea, in phase 3, normally if it is 100 patient or 300 patient, you have thousands. That means 10 times, 20 times larger patient. This larger patient we divide it, okay, then 1,000 patients, depending on the, so 1,000 patients, so in Karachi 200 patients, so Islamabad 100, depending on the population, depending on the etiology of the disease, we divide the number. It is not, it is number, it is the, we have to think the number very critically, from which area, which part of the area we do it, and which part of that, because with diabetes, what is the more prevalence area in Pakistan? Say Karachi, we give more number there. So less prevalent in say maybe somewhere, then we make less number. In this way, you have to calculate this number. There is another formula. How to calculate the formula? It's not your wish. Okay. In this way, the big difference is, as I told you, patients are different. So people here, people there, people there. So if you do the same compound tested in KPK, Lahore, Bhaulpur, Karachi, Multan, Sialkot, it all covers all the country, right? Only single center is good or multiple center is good. Same food in a party, same food. So you invited 100 people. Some people food acha 50% people bagbas. Same food, different people. Some people, those people like sweets. वो जो तुम्हारा food था ना sweet था, sweet बात कर रहा है. Those people don't like sweet. Both मीठा. So same food, different people, different comments. So if same people, same food or same medicine, same drug, it covers all over the country. It's better, far better. You give its idea. So okay, these people like very tea, 2 spoon sugar. So Karachi people they like Islamabad people look like 1 spoon. Karachi 2 spoon. Peshawar 3 spoons. Then how can you minimize it? 1 plus 2 plus 3 divided by 3. 1.5. This only way. Okay. Normally, oral glucose tolerance test, normally you have to give 75 gram glucose. It was not the case. It is 1.5 gram glucose per kg body weight. Okay, there is a debate. Someone told, okay, give it 50 gram glucose. Someone told, give it 100 gram glucose. At the time, the patient of IDF, International Diabetes Federation, he is from Bajaj. He make it, okay, make them, someone 100, someone 50. 100 plus 50 divided by 2, 75. He is happy, he is happy. Okay, sometimes they go, it works. So one is multi-center. And even, as I told you, patient is different. If you do some test, multi-center in here, multi-center and multi-country. 2 countries from Asia, 2 countries from Europe, 2 countries from Africa, 2 countries from different part of the world. It is deepest, depending on the, okay, one thing difference, that's why we need phase, because medicine is developed not only single country, it will, once it's developed, design, develop, planning for cover to the all but the world, right? Double blind technique. It means no one knows it. I am giving white, I am giving either green or red, patient is taking green and red. Double blind, and phase 2 is the single blind. Phase 2, single center. Phase 3, multi-center, multi-country. Multi-country may or may not, but multi-center. Okay, now the unfortunately is because you are in phase 3, so you are making medicine for the 7 billion people, right? But you are testing on only 100 or 1,000 people. That means you did study only a very fraction of people. So therefore, someone told me SNP is there, many things there. Some what the drug is very effective, very good in the phase 3. Then it is a market. After market, so normally phase 4 we call it long time toxicity. Long time toxicity. Okay, now. So after market, so the same drug, maybe one country it is safe, secure, but other countries patient cannot tolerate it. It makes from it, so as I told you, one drug, diabetes drug, Rezulin. So if you complain it, someone complain it, so getting by taking medicine, I'm not feeling better. Some side effects there, some death is there. It complains, then you have to withdraw from the market. Same is true after getting married. Sometimes optional question. If he or she become toxic, from same is true post market surveillance, long time toxicity. Is, as I told you, patients are different. Remember this word, patients are different. Okay, same medicine may or may not work together. The problem is, I told you, healthy subject. Then we came to the patients. We focus only patients say on single disease, but the patients, some patients, they have multiple disease, drug-drug interaction. It's very difficult. So therefore, those are pharmacy background, there's no better. So those are non-pharmacy background. Okay, so if you're taking multiple drug, so please read the leaflet, drug-drug interaction. Okay, so don't take 5 or 6 medicine together and take it at a time. This information should be transmitted. You have to take care for your mother, for your father, for your elder. So drug interaction. Read it carefully. हमारे पास मुश्किल है ना, फेसबुक के लिए 1 घंटा 2 घंटा 4 घंटे दवाई खाने के लिए ना टाइम नहीं, मुश्किल है, ठीक है. So post marketing, 40% drug withdrawn from the market due to long side effect. So 40% means almost half of the drug. 60 years, 18 years, it came to the market. After 1, 2 or 3 years, withdrawn. 40%, almost 50% drug. Patients are not. Patients are different. Medicine is the same. Somewhere it is working well. As I told in the early morning, one drug can save someone's life. One drug can may cause death to other people. You know him? So this figure, this photograph, anyone knows him? who's Atar Rahman. Okay, he was the founder chairman of ACC Pakistan, as well as he was the coordinator general of comstech. Okay, he told chemistry needs a new formula for success. Okay, he is 18 years time period, billions of dollars. A country like Pakistan, Bangladesh, it's impossible. So we have to think a different way. You have to think in different, you can make different plan. How we can do our own drug for our own population in our own mechanism. Isn't a lift in a lift? Building the lift, na? In everywhere you will see lift. In Pakistan, I heard and see some those are working on building construction. They make the lift by their own way. Okay. Sometimes I found building construction, you have to make some cement, sands mixture, 1 floor, 2 floor, 3 floor, you know, top by manually in this way. But sometimes I found something, some people had the cycle or motorcycle wheel, ring, ream, in the ream, they make it in this way. You need a pulley, right? But you can do it in your own way. In Bangladesh, I found the hospital, in the city hospital, this hospital, sometimes those are orthopedic patients, those are broken leg and hands. Normally, European country, they have to give some tractions, 1 kilo, 2 kilo, 3 kilo with the stainless steel weight. Because, but if it is do it in local hospital, if you give 1 kilo stainless steel weight, the next day you'll find it is nothing. Someone will take it. This is the reality. So what in the village people do it, very simple way. 5 kilo, they make it brick, 1 brick. A brick kitna hai? 5 kilo. So no one will take it 1 brick. हम तो चाहिए भाई 5 kilo ओजन weight चाहिए 5 kilo. Either it is loha, either iron, or stainless steel, or matti. My concern is weight. So some people visit from outside, they're surprised to see. They are local English. Safe, secure, don't take it. Okay. Then it's easy for them, for the patients. If he or she went to home, he can take that week anywhere. It's easy life. It is so sometimes you have to think it very simple way. So, ladies and gentlemen, energy hai. I'm giving a simple example. In Japan, so, you know, soap, sabun. So there is soap factory. There is someone in only one soap's cover is there inside. There is no soap. He or she make a complaint. This is real story. Then I got the soap, but inside
No, sir.
no soap. Only how to make it. Then the company made how to solve the problem. They make scanning machine. So after finished product, packet, finished product, scanning machine. Normally finished product at night duty, okay, scanning all the scanning machine. So at night duty, sometimes night guard or chukidar who is responsible for that to check all the shops inside shop, yes or no. At night duty is very difficult. So but what he did with scanning machine came bar. He spent a fan. In fan. So if it is soap, it will there. If it is no soap, it will. So simple thinking. It make the save millions of dollars. Continuous monitoring. Only 2,000 or 5,000 rupees. So $1 billion investment, 18 years time. It is very difficult for in developing countries. Therefore, I told this line, chemistry needs a new formula for success. You have to, our problem is different, our population is different. So we have to think our own way to solve our own problem, our own system, the own traditional system, local system. Why? As I told you, sometimes vaccine to give a good lesson to us. You have money, but the supplier is unable to refuse to give you. Even we did it in Dhaka, we give the ordered, we payment advance money, even we didn't get it. So for this, sometimes we have to take in the future our own way. If emergency comes, how to solve? How to solve our problem in our own way? Some people did, some country did, some Muslim country did. Some Muslim country, Western countries did their own vaccine. They didn't take vaccine from Western country. It's not also. Yes.
Sir, we always use drugs for budgeted purpose in case of communicable diseases. But in case of non-communicable diseases, we can use a drug for budgeted purpose.
Can you repeat one more, please?
Sir, in case of communicable disease, we use drugs for budgeted
Yes,
purpose.
yes.
But in case of non-communicable disease, we can use drugs for budgeted purpose or not.
So I'm not sure. I'm not sure. Anyone can comment on this question? Those are pharmacy background. And
बेसिकली आपकी कंडीशन पे डिपेंड करता है, आप कौन सी मेडिसिन लेना चाह रहे हैं और किस कंडीशन पे लेना चाह रहे हैं.
any other question?
सर प्रीक्लिनिकल और फेज 1 क्लिनिकल के दौरान जो फेज 0 है सर वो कब स्टार्ट होता है?
After normally clinical trial, before doing clinical trial, say you are giving some patient. So what I do? It is stuck in between. It is in 0 age in human, not in animal. So say we are giving 100 volunteers. Take 100 volunteers. For safe side, you get some idea. Or we are working on it. Our local surroundings, local small 3, 4, 5, 7 people. You get some idea. Then, because clinical trial is very expensive. Okay, so therefore we call it phase 0. It is in
starting of the phase 1.
Before starting the phase 1. Okay, you get some little idea. Sometimes if you invite someone, okay, so or if you want to cook something, okay, first we cook a small amount. Either taste it, even okay, then if you make a good big function, normally, so it passes different quality of rice. We make it biryani by oneself. Rice A, B, C. A, B, C. Then it's B is good, then process last quantity. So we get some idea. So same is true. So this 0, okay, some give idea who's kind of people, okay, who's kind of things or timing and things. At least you can get some idea to make the phase 1 more fruitful, more effective, better. Yes.
Sometimes drug got a good from the DA and it been in the market for 15 years, but sometimes it got bad again. So it's good or not, the DA should analyze the
There is, as I told you, to discover a drug, it takes
10 to 16
years. The problem is, so after we call it chronic toxicity, if you are taking medicine for a long time, it is almost impossible. Because we are giving patient this one after 1 or 2 or 3 or 2 years, we are observing for 2 years. For a long time, after heart happens after 15 years, these 2 years, it is cost billions. If we do a study 10 years, almost impossible. It's money wise as well as patient wise. No one will willing to be a volunteer or patients for a long time. Okay, so very simply, can anyone give me 100 rupees? Hey, If Japanese culture, nothing is free. Okay? Dekho, I ask you 100 rupees, many of you are willing to give 100 rupees. But if I give, can anyone give 1 bag blood? How many of you? Only one. Initially, okay, dekho. Okay, so I am sharing my own knowledge, own experience, expertise. Okay, taking money from anyone, very difficult. Very difficult. Taking blood from anyone, very, very difficult. And clinical phase, convince one patient to be a volunteer, convince a patient to be a volunteer for the clinical trial. It is almost impossible. Okay, almost impossible. We motivate him or her in different ways, these. So we'll do this one, we'll do environmental good condition, and we'll do in this time. We'll do all of your tests free of cost. This test, if you do in any hospital, in diabetes center, it will cost 10,000, 20,000, one single test, this one single setting. So if we do in our frontier every 3 months, 20,000, 50,000, we invest this kind of money free of cost, it will benefit you. In this way, we try to motivate. Nothing will be wrong. In this way, we try to make things. Okay, I'll quit thinking and let you know. He went home and it's the phone number phone. Next 3 days, this is the reality. This is the reality. No one willing to give the blood. Very few. Volunteer. Very difficult. Very difficult. But somehow we got it. So if he offers 150, at least we can take it. Then some people have some influence with someone. In this way, it shows going on. It's not easy. So 18 years later, we call it chronic toxicity. We don't know. It's impossible. By clinical trial, to understand this chronic is almost impossible.
Can I comment on this?
Yes, please, sir.
So, actually, the concept of pharmacovigilance this is actually addresses this question because the pharmacovigilance is a continuous process of the post-marketing surveillance, and usually when the adverse events are reported and they are like on certain threshold and up to certain magnitude, the authorities like FDA or other agencies like EMA, they take action and they can ban the drug. So there are examples that after the pharmacovigilance signal detection in different countries of some like adverse events which have been recorded in the patient, the drug has been banned or its use has been limited in certain conditions.
Like the case was with the dynamite, they resulted in deformment in the infants. During pregnancy, dynamite resulted in deformment in the children.
So recent this is actually a classic example of our pharmaceutical books like damage. So we all know that this can happen after 20, 30 years, but somehow you have to link it with the intended use. If you use in different conditions, there are chances that new adverse events will come up. So the agency's work is to see the pharmacovigilance data. There is like centralized data. There are centralized agencies and there are different national agencies as well, which keep a check on the adverse events coming up, and then they collectively decide that whether we should ban the drug as a whole or we should ban like certain indication of this. So there are possibilities.
Thank you, sir, for your nice comment and answer to the point. Thank you. Anyone? So at the last, this is this slide. Okay. So this is a poetry or Bengali poetry. So this is See very carefully. These two birds. Okay, one birds in the nest, another is the building. They are getting talk each other. Okay? So right side building, he is very happy. Okay, I am here living good condition, good building, air condition, this. And you are living there, winter season problem, summer season present, and rainy season have problem. Okay, then the bird, then old nest, he told, because you are there, you make them is little bit healthy. So sometimes if someone guest came, you will be sacrificed first. I'm here, my own house. Either it is here, either it is not building. It is my own house. I have some freeness. Even building. So the Malkin give you love and affection. Good food, good things. For that, they are giving this food or not for you, for their needs. If no one guest come, no meat is there. So pharmaceutical company like that. Okay, pharmaceutical company. So sometimes do this not for better on the health. Their money generating. They're not their money generating. Take it with this fever. Therefore, anything it is own. Your freeness to use it, modify it. Many things you can do it. But if it's not yours. And finally, this is the book. I searched many books, but this book is very interesting to me because everything is written very simple and very nice. Okay, so you can take this one. I can send. I have the software of the soft copy of this book. I can send Kazima to this book, or you can. I can soft copy, so you can collect. It make when you're at home after the conference, after the workshop. So many things written very carefully, very nice. So this is the end of my first session. So if you have any questions, anything.
So I have a little question.
Yes, sir, please.
So it was nice presentation, and you were thinking that some more words on the art part will come in your presentation. So as far as I'm concerned, I'm still looking for the art portion.
So after lunch.
You have mentioned
After lunch, after lunch. So after lunch, art and science. So initially it should be done here, but unfortunately it is due to time. So after lunch we'll do that. So Kadima, should I stop here or I can take?
I think we can go 10 minutes more. The session is like 12.
Okay. Okay. So Kazima is allowed in 10 minutes. Okay, please. Your second part, I'm starting now. Next time. Yes. One for. Discuss. Next slide, please. Yes. 12. Here on 12. Yes. Yes, sir. Another 10 minutes. We'll read draft design and development the art science. Okay, art is, we call it structure activity relationship, right? It is the combination of biology, chemistry, and statistics. You see, So, yes, everybody has mobile phone. How many people take photograph to this session? Okay, and you posted in Facebook, right? Normally what we do, we take 100 photograph, 10 photograph. Out of 10 photograph, this way, this way, this way, this way, in different angles, we make different photograph and which it seems the best. Out of 10 or 20 photographs, the best one we post in the Facebook, right? So, human is the same, subject is the same, all the changes in position, it makes different. So the art is, we call it structure activity relationship. So if one compound, say this is the compound, okay, this is the compound, and different side, it has some different group. If you change the group or rearrange the group, sometimes its activity increases, sometimes its activity decreases. Okay, sometimes. So this is the art of a drug discovery. So here this is called flavonate. This R1, R2, R1, R3, R4 many is there. So if you add A's, only A's, position 1, okay, A's, CH3, CH2, CH3, a different group. So initially this is the parent molecule. We got it active, lead compound. So I have some opportunity. So working only one compound, option 1, option 2, if we make different, if we add different group in different position, instead of one compound, we are getting multiple compound, 10, 20. So if one compound goes preclinical, clinical trial, say success rate is less. If 20 at a time, 10 compound goes similar compound, success rate is more. This is the art. Okay, molecular modeling and computational chemistry. Mostly the computational chemistry we do. So if this compound works in this way, if we change the position somewhere, either it is more good or no good. Very simple. Very simple. So we have to change this one, this one, this one, this compound. Normally what happens? Compound binds somewhere, the receptor is somewhere. So if we change the grouping, rearrange the group, this binding affinity, it sometimes increases. If binding affinity increases, you need low doses. This is the art. This kind of things we do in computer. We call it in silico study. In vitro, in vivo, it is in computer. The computer city, it's Silicon Valley in America. It's called in silico study. Okay, so this is one compound, right? We have different options. So this is our own experience. So protein kinase A dependent insulin-tropic effect of selected flavonoids. We find only one compound. It is very good. My colleague in chemistry, we did different computational study, and this is the left side. This is the parent compound. Are different group R1, R2, R3, R4. This one would make different one. Instead of one compound, we got many compound, more than one compound. This we got the compound, and this kind of compound we process it or synthesize it. Okay, we check it either my parent compound, other than parent compound, those we derive from the structure activity relationship, different arrangement, different combination, different permutation. This one either it is good or no good. We found yes, someone is work or better. Then initially you have one candidate. Now at least here we got 4 candidates. Working with 4 candidates, more safer, more better, success will be at least double. These are the art. Okay. Best example is, you know, penicillin. Penicillin is the drug. It is the antibody. Before discovering penicillin, people use salt as an antibiotic. Before in penicillin, some sulfur drug. Before sulfur drug, people use salt for as a antibiotic. So see it here. So this is the penicillins, right? This is the cephalosporin. This side is same. Only this is the side. This is the chain of penicillin, and there is some modification of this side chain. It makes cephalosporin. Okay, a small changes make a big changes. Sit here, synthetic penicillin. So this one, so up to this one, this is the same. Ampicillin, amoxicillin. But this one here, you see it here, 2 is here. Here, this H is there. Instead of NH2, this one there. Here, H is downside, NH2 is here, this one and this one. NH2, just make it reverse, and here OH group is there. Okay, only this is the arc. Okay, if you make some little changes, it makes improve your activity far better. Okay, another example is, you know, there is molecule, we call it, see it here, purine. Purine is alkaloid, right? This is the pyrimidine, this imidazole. It makes purine. Purine is a molecule of a DNA. Purine, DNA. So purine, it's become pyrimidine and imidazole. Pyrimidines, if it is pyrimidine, pi. What is the value of pi? 3.1, right? So in purine, that means number 3 position and 1 position, nitrogen is there, right? And 6, 7, 8. Now 7, 8, 9, 7, ACVN, N is there, 8, EIGST 8, no N is there, NINE, 9, N is there. Means 7 and 9, there is N. So 1, 3, 7, 9. But you see one compound, it's called, this is the purine, and this is xanthine. During purine metabolism, the purine to xanthine. So from purine to xanthine hypertension, ultimately it is last product is the uric acid. If uric acid is high, then there is a disease, we call it gout. Okay, it makes the high level of uric acid. It is high level of glucose, it makes crystal, that makes problem. So the allopurinol, it's a drug. Okay, here 7 and 9. Here only instead of 7, it's 8 position. Number of nitrogen position change, position, one position to another position. It makes a dramatic change. So xanthine, hypoxanthine, and allopurinol structure is the more or less similar. Only the number of nitrogen is the rearrangement. So in this way, it cannot work, cannot make the uric acid, etc. Finally, nowadays, future of drug discovery. Nowadays, I told you, it's computational study, computational thing. You can do millions of compounds in single click. Single click, you'll get it. But in case of AI, you can do billions of compounds within seconds. So now it is the era. Okay, without doing any test in the laboratory, you can do in AI. At least AI do sometimes better than you. No error. Now it is the era of AI. Okay, future of drug discovery. Now many companies already invest a lot of money opening this kind of thinking to do, because as you know, drug discovery is a very lengthy process. So it takes $15 billion. If it says only $1 billion, it's huge money. Money as well as time. Okay, sir, this is the art of drug discovery. This is the art of drug discovery. So the computational chemistry AI, this makes our life easier. It makes shorten the time as well as cost, almost zero cost. But you have to think it in very smart way. And we found that the computational chemistry and our laboratory work, it makes. Okay, it almost success rate is almost 20%. In our own experience, we found it. So it is active. These are the art of drug discovery and science in the experiment, in the test tube, rat, human. Yes, sir. You got the part? Now, this we stop here. So, anything from your side, any question, any comments? Yes.
In silico drug may be more effective, but whenever we design the drug and integrate in vitro or in vivo, that is not that much effective. That we have seen human protection studies.
We call it, some puzzle. It's still puzzling. Someone it's the number game. Number two is a evil luck. You are testing billions of compounds. Assume you find someone 10 compounds, no one is working. You are testing 100 compounds. Out of 100 compounds, one compound is working. Many things is not in your hand. Many things is not in our hand. And many things is here. Yeah? We call it tadbir and tadbir. You can try it. If it is not in here, you have to try it. It may or may not. Therefore, in silico, in vitro, in vivo, there is no correlation. There is no straightforward relationship. If you test 10,000 compounds, you'll get one compound. That's all I told you. In drug discovery, there is no prediction. There is no prediction. Getting married, thousands. Out of 1,000, you'll get only one. ठीक है ना? ये तो possible है. मैं एक ही से बात की. I saw only one. I got married with her. ये possible है कि नहीं है? It's possible. It's puzzle. Okay, puzzling. It's not very. There is no straight linear relationship. It's ups and downs. Therefore, I told you, drug discovery is a risky game. Risk is there. You may or may not. Okay, GSK, many pharmaceutical companies, they spend a lot of money and ultimately they disappear. Many years they did spend a lot of money in R&D. They couldn't make any drug in the market. Yes, sir. So today, this is the end of today's before lunch, inshallah after lunch. Yes, sir.
Traditionally, drug discovery heavily relied on systematic, almost formulaic approach. So my question is how some argue artistic institution and some creative approach to the molecular design to lead breakthroughs. But how can we leverage both contexts of SAR and potential artistic inspiration to accelerate the drug discovery with maintaining the scientific rigor?
So, SAR, structure activity relationship, right? So, normally when you do SAR, normally what happens? The compound will bind with the receptor. We then calculate. There is some point calculation. What kind of bonding there, and they make a score. What is the score of affinity in this way? So, I'm not a chemist. So, as per my understanding, so in this way, more you consider, more you take more time, then you may get better answer. Okay. Normally 2, 3, 4, 5, 6 only we considering a binding affinity, right? Only in the traditional speech. So if you do binding affinity, this is the one computational tool. Maybe you can check in other way in computational tool modeling. This is many different kind of modeling there. If you can do approach A, approach B, approach C, approach D, there is chances more in silico to in vitro or in vivo, chances more. But here the problem is that normally in drug discovery research, we are mostly students or teachers, faculty members, right? So you are doing for your MPhil or PhD degree. After MPhil, PhD, sometimes you forget your name, your roll number. So as I told you, there is a need for my second lecture. There is a need for academician and industry. There is a need of collaboration. My second part after lunch, we'll see it. Thank you. Thank you. Sit को दिया. Yes, student. So after lunch, you're sleepy or yes. His brother in online, they already joined. The second part of my lecture is biotechnology in drug discovery, from bench to bedside, from molecules to market. Anything you discovered, anything you identified, if you don't market value, it is meaningless. You spend lot of money, lot of energy, this. Ultimately, if it is no value, it should have some value. Otherwise, you cannot survive it. So, in the morning, we discuss the drug discovery, drug development. This is, we call it classical pathway. Classical or conventional pathway. Okay. So, the beauty of drug discovery, as I told you, it's a group of people. Chemistry, biochemistry, molecular biology, microbiology, biotechnology, right? When they share their ideas and views, many discoveries, initially it was from chemistry, later it's used for biology. For example, all the chromatographic technique, it is developed for the chemistry. Later, it is used for biology or biochemistry. So column chromatography, ion chromatography, all the things. But later, we can think the same principle. The molecules, the chemical molecules, chemical agent, either positive charge, negative charge. So our protein, either it has a positive charge or negative charge. Many things similarities there. Number one. Number two, so students, all of you are in biology background, right? Can you write your pen and paper? Can you write A? Right, देखा A. A, right? Someone write B. So if you think you are A, or someone who are A, raise their hand. Who are B? Someone write A, someone write B, right? Dekha? So think you are A, or someone is B. So tomorrow the task, initially you have some activity, energetic. Maximum activity will be A square. Your A square, someone is the B square, right? Initially you are A, someone is B. Your maximum, if it is A square plus, if you make, no, you want A square maximum, you are B square. If you make A plus B whole square, A square plus B square plus 2AB, right? So this is the best while in mathematics. This is the example, the grouping, teamwork. If 1 plus 1, you go. If you have 1, value 1, someone 2, the value, if you get together you working will be some additional value. This kind of 2, right? So therefore, the biology, that discovery, a group of people, A plus B plus C cubed, it will be more additional. So the things, many things discovered for the chemistry, but by discussion, A plus B whole square, it becomes more additive value. So SPLC. And this is choreography. Okay, this hanger sequencing, initially it was sequence for insulin. Later, this sequencing is done by DNA sequencing. Therefore, idea sharing, always discuss with the friends, even if it's stupid idea. Okay. So, idea sharing here very less, especially those are academician or those are industry. Bridging there both insignificant. So I'm giving some example. What is the role of academician and industry relationship? So naturally occurring protein. Say if you have some proteins in your body, cannot synthesize it, right? Protein, peptide. So chemical in plant materials, many things. The chemical compounds. So some of the body protein, so any disease I told you, it cannot synthesis more or it can overproduction. So if it is cannot synthesis more, then we have to collect this from animal sources, right? Then you have to sacrifice the animal. More expensive, number one. Number two, say one drug you are getting from cow, right? It is okay for you, but for the non-Muslim, though drug is drug, source doesn't matter, but some conservative people not use it. Okay, so same is true if some drug is derived from pork, for ours, you can use it, but some haziness, some uncomfortness. Okay, so I remember I did PhD in Japan. So I went there and here 2000 for me, they give a welcome party. I joined the party at night. They ordered pizza and top of the pizza, pork meat. Okay, then my professor told me, you can remove the meat and you can take the pizza. For them, it is very simple. But for me, it is impossible to eat. Okay, they don't know halal haram concept. Okay, then I didn't take it. So later, they for me ordered things, but they go halal haram concept. Then I it professor give me simple answer. Remove the meat, you can take the pizza. It is impossible for me. So this kind of barrier, real barrier. I heard many people died without taking insulin, either it is due to or it is written, but market available. So pot insulin, say there is no alternative. See, one country will import it. So there is no alternative. Then what do you do? So biotechnology give the best answer. Okay, I'm giving. So nowadays, it's many proteins, peptides. 170 peptides are in the active chemical development with many are more in preclinical studies. So here, biotechnology, or sometimes we call recombinant DNA to medicine. Combination and recombination. So if you see recombination means one molecule here, one molecule here, two molecules. If we cut and paste from cut here something and insert somewhere. Initially it was it is normal natural DNA. Now it is a recombinant DNA technology. This recombinant DNA technology, sometimes we call it biotechnology. Okay. One is pharmaceuticals. If pharmaceutical makes compound, molecules, drug. If the drugs is produced in living condition, it is called biopharmaceuticals. Bio means bio, living condition. So some data. So how the biotechnology drug, bioproduct is increasing more and more. So this is the biopharma industry. Biopharma industry repeat very good, especially after COVID. It's very rapid jump. COVID. So normally in developed countries, US, UK, this country, they're most focusing on biopharmaceuticals. But in developing countries, we're focusing on traditional medicine, generic drug. Products of modern biotechnology. More than 60% of biotech companies in US are involved in pharmaceutical production relating to the drugs developed for the medical use. And 1982, first drug, biotech drug, insulin. Okay. So as I told you, recombinant DNA technology allows DNA to be combined from different sources. In this way, we can clone genes, and by doing so, we can do many things. Sit here. So round one, this plasmid, right? Okay, plasmid. So sit here. So this is the plasmid DNA, right? Okay, we cut somewhere. We cut it here. Plasmid DNA, it called in bacteria, there is some special DNA, close circular DNA, round shape. Okay, if we cut it, there is a gap, right? In this gap, I can insert my gene of interest, So if it is initially it is 1000 base pair, now it is we add this one, now it has become 1200, 1200 base pair, right? Now the plasmid contains its own gene as well as our extra gene. This plasmid, we call it recombinant plasmid. This recombinant plasmid, we call it technology, normally plasmid. So if this plasmid we insert in bacteria, then bacteria will produce its own gene as well as this gene. Recombinant DNA, we put this recombinant into the bacterial system, living condition, the bacterial system, and put this is called my target gene. Ultimately, production is the protein, right? It called biotechnology or recombinant DNA technology. By using it has more advantage. Advantage is pharmaceuticals and biopharmaceuticals. For pharmaceuticals, you need a big industry. For biopharmaceuticals, you need a smaller space. And only overnight, one bacteria to two bacteria, how long it takes times? One day or 3 days from one bacteria to two bacteria? Minutes, 20 minutes. 20 minutes. Doubling time, 20 minutes. That means if you grow bacteria tonight, tomorrow morning you will get billions of copies. That means overnight is enough. And small fermenter is enough. So it is the beauty of biopharmaceuticals. Okay, and target oriented. So it is inserted the target gene. The beauty is that your target protein is expressed more and more. So purification normally in plant 1000 compound, only one compound. Purification rate is very slow. Yield is very low. Here yield is 50%. Yield is 50. If yield is 50%, then it is normally small sources you will get more amount. If yield is 50%, the purity possibility purity is very easy, and purity efficiency is very easy, 99.9999. Because this is the recombinant DNA technology. So using recombinant DNA technology, it is used for health, medicine, agriculture, medicine. So if you think in maize, okay, so you're taking some things, banana, right? So if we add some genes other than banana genes, you're taking banana, you're taking other things. Golden rice. In rice, they inserted vitamin. Okay, you are taking rice, you are taking vitamin others. This is the advantage of recombinant DNA technology. Okay, so then it is very easy. Okay. So the production of anything in living condition is biotechnology. Biotechnology, molecular biology is the subject, and biotechnology is the real application of this molecular biology, right? So using molecular biology or biotechnology can be used for betterment of health, production of better quality of food, better quality of medicine, and many things. And this is the new era of. The big thing is that during COVID, the vaccine was developed, COVID vaccine. It's a biotech drug. Biotech drug, and number one first biotech drug is the insulin. Insulin. And very interesting, insulin got 3 Nobel Prize. 3 Nobel Prize. One, someone discovered the insulin, Benting and Best. He got the Nobel Prize. And Sanger, it is amino acid sequence, you just sequence the amino acid. He got the Nobel Prize. 3, radioimmunoassay. Now we take one assay system, radioimmunoassay. You know it? You know ELISA, enzyme linked immunosorbent assay. So if something is the milligram quantity. So protein is the milligram quantity. If you put in the hot water, boil it, if you starve it, it is protein. It's milligram quantity, gram quantity. If something is gram quantity, you can test by your own open eyes. If it is milligram quantities, anything in your body, you can do some laboratory test, spectrophotometer, photometer, by taking observance, you can measure it. Okay, but if it is less than that, nanogram quantities, spectrophotometer cannot measure it. Okay, this only option is HPLC. Okay, high performance liquid chromatography. If it's less than nanogram, picogram, HPLC can't do it. Then radio immuno. Okay. So the advantage of radio immunoassay is very accurate, highly accurate. The disadvantage is it's a substitute technique. No one can do it. Not everyone can do it. You need it. So using radioisotope is easy, but discarding the radioisotope is very difficult. You have to, once you pass the radioactive compound, you need licensing. Number one, using units explain training. I can understand. I work radioisotope. I got 3 month training for working 1 month. Okay, and for radioisotope is risky. Okay, so using radioisotope, every month you have to check your blood, either your contamination or not contamination. If you're contaminated, certain stage, some complication things is. So our insulin level is nanogram, picogram. So it's impossible to measure. Only one solution is radioimmunoassay. Okay, someone Dorothy Hopkins. So he measured the radioisotope using the radioimmunoassay. She measured the insulin from biological sample. She got one Nobel Prize. A single molecule, okay. So size doesn't matter. Okay, single molecule, a small molecule. Okay, so in Bakai Hospital, diabetes hospital, there's a project, insulin, my life. Okay, because those are type 1 diabetes. Those are type 1 diabetes without insulin. For survival, they need insulin. Okay, for type 2, without insulin, type 1 diabetes will die. But type 2 diabetes, without insulin, they will not die. For type 1 diabetes, insulin is needed for survival. For type 2 diabetes, insulin is needed for the management. Okay, because all over the world, type 1 diabetes, type 2 diabetes, type 2 is more. And Allah Ta'ala give a special consideration, these are Asian countries. Here, type 1 diabetes is very less compared to the Europe. Okay, in 1 lakh, in Pakistan, only 1. In Europe, 1 lakh, almost 2200. Once you get the insulin, rest of your life with insulin. So I did give this project, my insulin, my life. Okay. Okay, the discovery of insulin. I told you in the morning, John Dr. Benting. Okay, sometimes normally the teachers, students, academicians, those are teachers, they teach, but business, they're not businessmen. They're always crisis in money, right? The Benting is a medical doctor. He graduate medical doctor. So you told many students in the morning time you are unmarried after MPhil or after PhD. So if you don't want to marry, your father or mother will force you to get married. This is the reality. So he want to Dr. Benting, he want to get married. So then he needs he make a chamber, but chamber of patient is not too much. Then for getting married, sometimes misery, some poor condition is good. So what he did, he went here and there. Okay. So and then he got in the university and got some research work. He needs some assistant like that, assistant to work on it. They give the position and $10 per week. Okay, he accepted it. Okay, And he okay, he discovered, he purified insulin and changed the life, changed the life of many wives. Okay, all the world, it is. Okay, simple thing. Okay, therefore, if you got those students, if you are get immediately after getting your graduation, if you get job, all and good. If not, maybe something good is there. Okay, so if you got the job, you can change your life, maximum your family life. But if you don't got your job, you may change the whole world. Okay, don't be frustrated. If you got job, good. If you don't got the job, more good. You can write poetry. Poetry, you can write drama. Okay, so now anyway, so the insulin study, so here, after initially, as I told you, insulin was purified from animal sources, either from cow or from pork. The human insulin, pork insulin, and beef insulin, the insulin has 51 amino acid. And from human to pork, 2 or 3 amino acid changes. Human to cow, 3 or 4 amino acid changes. But if you got the insulin from pork or if you got the insulin from the beef, it working very similar. Not 100%, but very close to 100%. It working. It manages the diabetes. Okay, as I told you, they work on it. They work from isolated and purify the insulin and give a dog, and dog cut the pancreas, that means it makes nothing, and they give their extracts. Okay, and the crude extracts, it is in Melbourne. One young man was type 1 diabetes, and he's supposed to die after some times. Then the crude extract, someone is in hospital, he's working in hospital, then okay, please give the injection, crude extract, not purified one. Give it, it's miracle. The young child, it is supposed to die. He becomes more younger, active, dynamic. But when he died later, but the duration is supposed to die a few days later, a few months later, but he survived many days. So this is the history of insulin, right? The problem is, okay, so you have to sacrifice many animals, number one. Number two, religious constraint. Okay, so this problem is solved by the global insulin market. Okay, there's lots of market all over the world. Okay, this is the insulin molecule, right? There is a chain. Okay, this is a chain. This is one is 21, another is 30. 51 amino acid, right? So 50 amino acid there, and there is some sulfur, okay, cysteine molecule. Then some cysteine molecule, disulfide bridge is there. So if you destroy one disulfide bridge, the insulin activity will be gone. So this is the molecule. This molecule, very interesting molecule, 51 amino acid molecule. It is, they told you, the gene was cloned and inserted to the plasmid and put it in the living condition, it gives insulin. How it happened? See it here. Okay, recombinant DNA gene, science to business. Okay, normally we are molecular biologists, academician. What we do? We publish paper. So the pharmaceutical company, they use 20% of their budget for research and development. Okay, So they publish papers. So normally those are working on research and development, pharmaceutical company, they read articles, journals. So he's the man, he's the manager of a pharmaceutical company. He's reading article. So he saw the article recombinant DNA technology. He cut it, insert it, recombinant DNA technology, new protein. By reading one, okay, he published the paper. And he's the industry, from industry side. He read the paper and tried to get an appointment from the professor. It is very difficult. Professor is very busy. He gives 10 minutes time to him. So please, next month, this day, this time, 10 minutes. Okay, they came here, they publish paper. He thinks if we put something there, and he personally think insulin. We put insulin gene there, it will be recombinant insulin, huge market all over the world. It saved many lives, and the religious constraint, it will be removed. Okay, so 10 minutes discussion, it becomes 6 hours. And same table, one pharmaceutical company formed by Genentech. Okay, 1976. So therefore, academy and industry people. So you have some research idea, but commercial idea you don't have. Marketing side, we are very poor, university person. But industry people, they know it. They can predict it. Okay, by collaboration, cooperation, as I told you, A square plus B square. When they make together, A plus B whole square. Okay, it made some additional value, some additive value. In this way, recombinant DNA technology was born. Now, recombinant DNA was born, but it is new. Biotech company was formed, and their first biotech drug was the insulin. First drug is the insulin. Now, so as I told you, this is the insulin molecule, right? So if someone made something, everybody want to do it. Okay, someone made insulin, right? Then someone think we have to do better insulin. What they did, 51 insulin, they did some computational study. We call alanine scaling. Replace each amino acid by alanine. So if you replace this alanine, replace different amino acid with one amino acid with another, so maybe the activity enhance. Okay, so someone what it did with this amino acid, there is a side chain. In side chain, there something, so that it will be more efficacious and it will be more stable. Therefore, in market initially to insulin, now we find rapid acting, long acting, we call it insulin analog. It's nothing. There is replace either one or two amino acids somewhere, or in the side chain there's something. One is insulin, another is the insulin analog. Nowadays, the biotechnology goes in such way that, so it's very difficult to take insulin, right? What they do, the human insulin gene, they give in cow. So we're taking milk, you're taking milk as well as insulin. It is already done, but it is not commercial yet. But the problem is, but insulin injection and insulin oral. Okay, at least if you take 2 times milk, it's okay. Or if taking injection is not friendly, not user friendly, taking milk. So initially, it's very easy. So insulin, sometimes people make some recombinant technology. Insulin is potato, insulin is tobacco, but it was not successful. Let's see, it's improving and improving, lot of improvement there. Okay, so one is genetic engineering. Okay, see here, the plant is green. Okay, so in the Islamabad, okay, sometimes there is electricity crisis, right? So there is a concept. At night, some insect inside in the dark place, it eliminates some light. What is the name of that? Hive light. Okay, so there is an enzyme which called luciferase. Luciferase. So main lighting is the luciferin. So people are thinking if this luciferase gene is isolated, inject or make some plant, give in the plant contain the luciferase. At daytime it was the normal light, and at night they will eliminate the light. The whole street will be enlightened. Okay, no need any electricity at night. Okay, people are thinking maybe if there is no electricity, something will be happened. Then at your home, you purchase, instead of purchasing bulb, you purchase 4 plants here. At night it will be light. Maybe our next generation, no electricity. Everything. Purchase tree, take care of the tree, but at night, at day it will get in oxygen. At night it will give lifetime. This is the advantage. Okay, so this is my own experience. So during my PhD, I purified almost half a dozen of recombinant protein. So using this protein, recombinant during protein, so though I work on diabetes, I have mind. So is it possible to purify insulin in Bangladesh? So in between my PhD, so I came to Dhaka and discussed my process. And so this is I did all the column chromatography, the techniques needed for biotechnology for recombinant DNA technology and production of recombinant protein. I know it. I did it. And after coming to Dhaka, so there is called ICCB, International Center for Genetic Engineering, Biotechnology, Italy. Okay, they have some biotechnology center, and there is a biotechnology development group. I communicate with them. I want to do insulin in Dhaka, Bangladesh. Possible or not? They give me suggestion. Okay, they give me letter. Yes, you can do it. You can come here. We will get training and laboratory scale. You can do it laboratory scale, and you can make it in the large scale. But you have to attach to some pharmaceutical company. And they told me, okay, and the best example is the Argentina. Someone came here, we got the training, and recombinant DNA technology. The recombinant DNA, it is inserted in the bacterial cell. Normally, they provide the cell. Just you come country, fermentation, do it fermentation, to purify this. But at that time, it is too early. Someone asked me, why I want to do it? Because at that time, recombinant DNA technology is not familiar at the time. So no one take the risk. Okay, it's unsuccessful. Anyway, I think at that time. So as I told you, column chromatography, ion exchange chromatography, affinity chromatography, this is the main chromatography used for the protein purifications. So another big thing is the antibody. Antibody is a protein. Antibody is protein. Nowadays, monoclonal antibody, it's a treatment using it's a protein. Insulin, you are making insulin, recombinant insulin, same is true. Anything you can insert it, either luciferase or monoclonal, anything you can insert it, you can express it, purify it, and it is the product. And nowadays, insulin is a big market. You have to put many big market, many biotech drugs available nowadays. Monoclonal antibodies, immunotherapy for cancer patients. Okay, those all are using this biotechnology. All are using biotechnology. Monoclonal antibody, normally it's called hybridoma cell. Okay, so those cells collect the spleen, this cell and hybridoma cell, that means it immortalized. Whole life, it will be this one. This monoclonal antibody product the antibody. This one by using the different contact technique, you can use it. Okay, one of my friends, there's a big company it's called Capcom. They use that 30,000 enzyme commercially produced. One of my working there, one of my friends, monoclonal antibody. So many monoclonal antibodies available in the market. all the monoclonal antibodies for the betterment of health, especially betterment of the cancer patients. It is by the biotechnology. Initially, biotechnology in the veins, right? Veins mean working veins. Normally in the laboratory we call it veins. Now the monoclonal antibody or insulin, it is in bedside. Normally if you sleep, bedside there is a small table. We put medicine there. So you see, biotechnology recombinant DNA product, initially it is veins, now it is in bedside. Veins to bedside. So that you need medicine, no need to go here and there. Take the medicine. Veins to bedside. Another is nucleic acid therapeutics. So, you know, nucleic acid, RNA, DNA, mRNA, these are the most important is siRNA. So anybody has idea about regarding siRNA? Anybody has any idea?
Yeah.
Okay. A small interference RNA. A small interference means, so normally if you see this one like this one, right? So it is 19 to 21 amino nucleotide base pair, there is overhang elements. So it normally DNA, RNA, protein, right? So if you add a small interference RNA, it is single stranded RNA. Okay, this siRNA is the double stranded RNA, not single stranded. So if DNA, RNA, protein in mRNA, you make a complementary siRNA, then it binds to the mRNA. Okay, if it is bind in the mRNA, the mRNA to protein will be switched off, either 100% or 0% or 50%. If it is exactly matched with mRNA, 100% inhibition. Okay, normally this siRNA for 100% matching, we have to make it by laboratory. Okay, which one is the mRNA? You know the sequence, you can make it. But the siRNA is also available in the living system. Another is miRNA. Okay, miRNA. The siRNA, it matches partial matching with mRNA. But the miRNA, it matches RNA, mRNA 100%. And it binds many places. In this way, it can suppress the production of protein. Okay, this kind of technique. So mRNA based therapeutic. Nowadays, many things that cell therapeutics. Engineering the next generation of cell-based therapeutics. Then nowadays we call stem cell transplantation. Okay, so the cell and stem cell, there's a difference. The same cell, normally in one cell, is divided in the same cell, right? What is the beauty of stem cell? Okay, say A to A, normal cell, 8A, 2A, 2A to 4A, 4A to 8A. But in stem cell, for A, next is A, anyone, A, 2A, A plus B. In this way, it can differentiate as their needs. So this is the beauty. You're giving something and output is other thing. So therefore, if you think it, you need some, see this cell can produce something, this is your medicine. We're taking medicine orally or injection. If this kind of medicine can produced by the cell. This is a drug. It's called drug of biology. And open innovation, a collaboration between academia and the pharmaceutical industry to further leverage drug discovery, expertise, and assets. So, as I told you, the study of insulin, okay, so someone should come forward, okay. So, if someone should, either industry people or academic people, sit together, it will be more beneficial for the society and for the human being. Okay, nowadays, okay, though many drawbacks is there, even in Europe, now our drug is there, some pure components also available. Some pure, it is more expensive. We call it personalized medicine. So maybe later in this kind of medicine or biological drug, it may be available for the personal use. So the antibodies, it's a big future. Okay, the production of antibodies is not difficult. Okay, not difficult. And it is the promising side. So this is the drug of using living condition or somewhere in using biotechnology. So the beauty of this biotech drug is, the advantage of biotech drug is, in a small space we can do it. The disadvantage is that, what is the disadvantage of biotech drug? Some disadvantages also there. It's expensive. Number one. Number two, in normal generic drug, paracetamol, this kind of drug, you can put anywhere. But this kind of drug, you have to take special attention from production to at your home. You have to make some cooling system. Okay. So if you don't maintain the cooling system, it is biotech drug, but the efficacy lost. Therefore, yes, dear participant. So the workshop, it will be good for your practical life. If you want to purchase for your father or your mother, for anyone, insulin, make sure you are purchasing insulin or it is not insulin. It is written insulin. Okay, but it is already during cold chain maintenance, destroyed activity, it's placebo, nothing. So therefore, once this kind of drug you will be purchasing, please be careful. Please be careful. Sometimes we found our center, that means many people are giving insulin, but it is not working. Okay, then later we came to know that the percentage in this kind of drug is somewhere. There is no cold chain maintained. The percentage, and even they don't know this kind of insulin, you purchase from someone, they didn't inform me that you have to maintain it in the cold chain. So this is some advantages there, some disadvantages there. Evolution of therapeutic antibody discovery technologies. Okay, we call it therapeutic antibodies. Nowadays, the trends is going to antibodies. So antibodies, normally the COVID-19, we give some vaccine, right? It is protein. It is antibody. It is antibody or not? No, it is antigen. We give antigen, so it produces antibody. So antigen, antibody, all are protein. Okay, so these are things, antigen or antibody, all are proteins. These are making and making purification, extraction, all the using is recombinant DNA technology. Recombinant, this is the only, this is this recombinant DNA technology power. How powerful it is. Using the recombinant DNA technology, the COVID vaccine was successful. It came to the market. Before marketing, all over the world, the people, it's mandatory to take this vaccine. So we all are carrying recombinant DNA technology product in our body. From veins to body inside. You have biotech product inside. Almost all you have. Recombinant DNA technology part of this product inside. So you can't avoid it. So biotechnology makes all life easier, especially the agriculture product. Okay. So finally, as I told you, only drug, right? Chemical agent, this is called drug. Biotechnology is a drug. So he was the man, he was the president of IDF, International Diabetes Federation. He told one thing, for diabetes, education is a treatment. So understanding the mechanism, understanding the things, what things before meal, after meal, timing, this kind of knowledge is needed to make it good health. Doctor can give prescription and only medicine. You have to take care of yourself. We found that the more educated people, they take good care of their health. The purchasing medicine, it is only one thing. We found that many people don't take medicine in time. They take medicine in their own way. Normally, doctor are MD, but patient MD square. They make their own prescription. Okay, so education is not a part of treatment. It is the treatment. Okay, so I give sometimes public awareness lectures here and there. So during diabetes, so I told, okay, avoid Coca-Cola. Coca-Cola, I told you. I did Coca-Cola, Chola. Okay. Coca-Cola or Chola, what is the name? Everybody likes it. These four, male, female, all the religious people, everybody likes it. So, and every corner of the part of the world will find Coca-Cola. Where there's a Coca-Cola, there's a diabetes. Someone asked me the question, sir, instead of Coca-Cola, can I take Pepsi? Okay, that means certain amount of knowledge is needed. Okay, Coca-Cola means not Coca-Cola, it's called Coca-Colization. Any. So, young man, so this is the book. Medicine, there is no relation of medicine and health, medicine and happiness. It is healthy mind, young mind. So, age, young, it's not a number, it's your mind. Some people at 20, some 40, they behave, the mindset is 60. Some people is 80. I found Professor Athur Rahman. He's, mashallah, many years, but still young. Mentally, he's young. Okay. So this is the book. So you need a sound mind, sound body. Health is only one thing. Medicine, only one factor. Other medicine, many factors there. Okay. How you take the medicine, how you behave yourself. If you're happy face, happy mood, it's also medicine. We call it diabetes, we call it laughter therapy. If you smile, laughing, it's a medicine. And it works for diabetes. Sometimes in Europe, UK, look at dancing therapy. Dancing is a therapy for diabetes. No need to go to the gym, dance. Okay, that means all are medicine. Most important is doesn't matter. Okay, in here, be young. And this is the book Ikigai. It is in Japanese book. Okay, the Japanese secret to long and healthy life. Normally, here at 60 years or 70 years, we are old, we are dependent on someone, right? In Japanese people, even that live 100 years, they've been dynamic, young, energetic. How to do it? They found one thing, how to make happy. Which thing you want to do it with happily, do it happily. If you want to do anything, if you cook it, do cook happily. If you sleep, enjoy your work. So it will make happy and healthy life. So you want joined my workshop. Thank you very much. Enjoy it. Don't take stress. Mood. Happy mind. With this, so now it is time for yours. If you have any questions, last one. Think it from today. Think how you can do biotechnology to business. Always money. So if you have money, you can buy honey. Okay, biotechnology to business. How to make DNA? DNA means dollar. It's recombinant restriction enzyme. They have DNA. Okay, enzyme to euro, and how we can the research you can convert to rupees. Someone asked me, he is working on collagen, right? Collagen. I told her, okay, make a face wash or cream. Use it first by yourself. Surroundings. If it is active, not toxic, open a Facebook page. Cardiazum products, a product of cardiazum, or what is your name? A product of Gulro. Young for lifetime. Young at the end of life. So, bas, Facebook boosting karo bas. If you need anything for our site, for comstech site, we are ready to help you. Yes, sir. So, for my part, this is the end of for my part today. Now, you have any question, comment? I'm in open discussion now. Yes, sir. जोर से बोलो भाई, खाना खाया कि नहीं खाया? Yes.
Yes. So, we have already seen that in developing countries, the age of developing diabetes is after development, but in developing countries, it is life that. So, what do you think that the cause of that is? Is it genetics or lack of environmental development?
Ecogenetics. Okay, ecogenetics means it is genetics as well as environment. A bullet, no, pistol. In pistol, already bullet is there, right? Bullet cannot do anything. You have to trigger it, right? This triggering is called it environment. So if your gene is there, chance is 25%, not 100%, but remaining 70%. If your gene is there, chance is 33%. Okay, if you consider triangle this one, okay, genetics here lifestyle, here environment, 33, 33, 33. So if you have got from genetics 33%, 66% you are safe, but this is on the lifestyle and environment. So their lifestyle, our lifestyle is different. Normally, in here I found here there normally in Pakistan, Bangladesh. So many Japanese friends came to Bangladesh. They stayed 7 days. They complained me that 7 days we took the same food. Then my wife made phone call. many places. They told me, no, we make chicken, we make mutton, we make beef, different food. But pattern is different. In Japan, they taste the meat, only salt. It is meat. But here in Bangladesh, Pakistan, India, we don't make cook. We got the taste of masala. Piyaz dalo, haldi dalo, nimki dalo, adrak dalo, rosun dalo, darchini dalo, tejpata dalo, curry masala dalo. So ultimately, taste of fish or taste of meat, this is here. You get the taste of masala. So they are using less oil, less salt. Okay. So we are using normally here, but in our country, every day we are taking rice or bread. Year and year. 60 years we are doing same food. No variety. Okay, we are taking we have a plate system, 1/4 raw materials, 1/4 rice, 1/4 carbohydrate, 1/4 protein, 1/4 lipid. We are taking rice, bread. Food habit we are taking same food year and year. Number one. Number two, late night eating, late night sleeping. Normally, European people, you know, Japanese people, European, once they sleep,
Early to bed.
early to bed. Okay, early to bed, early to bed. Okay, normally they sleep earlier. Normally the alpha sleep, the alpha level sleep. So 9 p.m. to 2 p.m. it is called alpha sleeping. The best sleeping mode is one. Normally Pakistan people start sleeping after 2. Late night sleeping, late night sleeping. I want that. This is found this oil. This will not this one. This is the cause. That is okay. This is some early to. So many things positive. Normally, and this mosquito coil. Mosquito coil. So one coil is if you take tobacco, you and surrounding people will be affected. In mosquito coil, whole environment will be polluted. And one coil is dangerous more than 100 cigarettes. Okay, so therefore, this is few, not all. This is the difference between one is food habit. So yes, there is a risk calculation for diabetes. If you are 40 plus 1, less than 40, 0. If you are Asian, 1. That means by born, we got 1 3 point. Asian are sometimes called the 80% diabetic patient in Asia. So I found one thing, it is philosophical problem. So my body weight is 70 kilograms, right? My leg is 3 feet, right? 70 kilograms, 3 feet. टांग है जीवा कितना वेट है 100 ग्राम and 100 gram 2 inches. We are focusing only 100 gram and 2 inches. This is the basic difference. Okay, we consider only time. We cannot consider requirement. Okay, so this is the reality.
So someone is asking question, I think they are online.
Yes, sir.
And on the Zoom. Yes.
What kind of question? That is your property. to effective. Can you make it more? A nice session. Question. What is the question?
The hand raise.
As per. Yes.
I think you are appreciating on.
No. Yes, online students, online participant. If you have any questions, next 15 minutes for you. Please ask question on chat box. You can only ask they can speak. No. Okay, we have only option. You can write your question or comments. Okay, in your chat box, I'll try my best to answer your questions or comments. Please. Next 15 minutes. Those are participant here. Please allow me 15 minutes. Okay. Please. This is very interesting. Please share material if possible. Please ask question. So if possible, it's dependent comstech. Okay. If it help, I can give it, no problem. So then I can give my presentation to comstech. Okay, if someone need it. Anything more? So if someone meets the session of your friends, okay, from of your university or institute, so I found many things, someone is absent. If you find your surroundings, your colleagues or your students, okay, please inform it, right? So that they can catch the train tomorrow. Yes, as I told earlier, the lecture was very interesting. Please share material if possible. Yes, it is 100% possible. Please communicate with comstech people. Yes, another 5 minutes. Students, if you have any question, if you have no questions, we can stop here. What is the important step in drug development? Yes, Mohammad Faisal. Okay, what is the important step in drug development? So, for simple, all the steps are important. All the steps are interconnected, and all are interconnected. Most important is to toxicity. It is in vivo, in vitro, or human. Most important is toxicity. It should be no toxic. It should be safe. Either it is effective or not effective, it's later. First, it should be, mandatory is, it should be non-toxic. Non-toxic or minimum toxic. Minimum. Yes, sir. From here, any questions, any comments?
Sir, I have a question.
Yes, please.
Sir, you said that the diabetes is more common in Asian countries. But I have read a paper previously that diabetes incidence rate is less common in underdeveloped country as compared to developed country, but the crude mortality rate and the age standardized rate is higher in underdeveloped country as compared to developed country. So would you like to comment on this?
Yes. In underdeveloped countries, so in Europe, America, they have health insurance. with this health insurance, every year, 2 times, it's mandatory to do the routine test. Every year, you have to check your blood glucose. 100% people under diagnosis, either he or she has diabetes or not. Every year, you are diagnosed 2 times. But here, people are not go to hospital either I have diabetes or not. Mostly, people admitted hospital or go some other reason, then doctor advice, please check diabetes. Okay, with the advice of doctors, they check and found the diabetes. Okay, they were diagnosed diabetes either day 0 or day 3 month. But here, diagnosis, it is sometimes 1 year, 2 year, 5 year, even 10 years later. Normally, we went to hospital or doctors at the very end of stress, very severe condition. So we call it diabetes. But due to diabetes, mortality, death is 0. Due to diabetes, all the death is related to diabetic complications. Diabetes, neuropathy, nephropathy, retinopathy. So if you diagnose diabetes today, you can manage your diabetes whole life. You can manage your life. But if you don't you diagnose 5 years later, 10 years later, that means when you diagnose diabetes, your diabetic complication is already there. Okay, so there our prevalence rate is less because we are not 100% people are under screening condition. There are 100% people are screening condition. So we are getting the prevalence with just assumption. If you do more in Pakistan, 26% people have diabetes. 26 means every 4 people there is a diabetes. Your question answer is mixed. It may or may not true. Okay, normally 80% population is living in Asia in pains because in India 1 crore diabetes, China 1 crore diabetes. It is a number game. Almost all people, the people all over the world, maximum people here. India, China, Bangladesh, Indonesia, Pakistan. If you see the democratic people, almost all people are here. 6 number, more the number of population, more the diabetic. If you consider 2 cents, prevalence, even Pakistan is 26%. If you diagnose 100%, you will got the true figure. It is not possible. Exactly. Sir, what are the input mistakes? Sir, another question, please. How to then we can solve challenges in Pakistan for drug development? Yes, madam. Young man, hot blood. You can think. Small thinking, think small, simple. Think big. Start with a small thing. This is the simple solution. Think it. So biopharmaceuticals or pharmaceuticals, it's $1 billion. So think it a small product. A small product. Those are used in traditionally used for any disease or many disease. This kind of herbal products, if it is edible, okay, people are using it, or some kind of portion of your country use it. You can use it as a drug. So we are talking about the synthetic drug, chemical drug, but there is another thing is Unani drug, herbal drug. Okay, this is also more effective. So in chemical drug, after making the drug, we make the clinical trial. But this called traditional medicine, we call it traditional medicine. It is already we are in clinical trial. So those are available, those plant materials or those herbs available, affordable. You can choose 1 or 2, and you can make herbal product, okay, before going to the real pharmaceutical product. It is the very shortcut, and that's going to be doable in Pakistan. Yes.
Sir, here in Pakistan, we hardly see any collaboration between industry and the academicians, with few exceptions. I want to know about the Bangladesh status in this regard.
पक्का मुसलमान का एक ही बात this is the story. It's normal. Don't mind it. It is that our Muslim groups who are very deep debatation. Okay, don't mind. Real thing we are unable to focus. Okay, but unreal thing, how to think? We have spent a lot of time here. So collaboration. देखो collaboration तो बहुत दूर का बात है. हमारे तो भाई भाई ना, two brothers. Before marriage and after marriage, two brothers, two wives. Collaboration, it become collaboration, So this is the sad part, honestly speaking. You know, someone should take initiative. You have to take initiative, sacrifice. Someone should be sacrificed. I want to sacrifice your mind. Or number one. Number two, I want to start. Everything we can do by ourselves. Everything we want to do ourselves. Okay, so I was telling last story in Japan how they develop. When I was there, they couldn't understand English. Then with mind, how they develop economically. Then I found that. So it is comstech, right? If I ask, where is the room number 40? If I ask Chokidar, where is the room number 402? He will tell, I don't know. If you want comstech, you will ask. Any people ask, where is comstech? I don't know. This is how they develop. The focus. Chokidar also focus to take the gate. 100% efficiency there. If you don't, room number 2, 402, third floor, then you ask how I can go there. Then please can you share and show me. Then your attention will be diverse. Definitely focus. जिसका काज है ना, focus. Therefore, they develop. They will be focused. Okay, from today, answer is I don't know. If it is not, so what we are doing, as I told you, 9 to 2 p.m., 9 p.m. to 2 a.m. This is the alpha sleep. Normally, European people, American people sleep that time, and we start at 10 p.m. or 10:30 we take dinner, then start Facebooking after 2. We destroy our life by ourselves. Okay, because they implement free, Facebook free, so that sometimes you will be dependent on medicine. They will develop medicine and sell to you. Okay, they give intention free. If you ask the father or mother, give me 1,000 rupees, they'll give it. If you ask, give me 1 lakh rupees, they'll give it. 10,000 rupees, you'll have many questions. So they are giving free everything. Why? It's more than your father. Think. If you change, you can change. Otherwise, if you start late night eating, late night sleeping, you cannot change yourself. So sometimes you cannot change that more. You cannot change the channel.
Question, sir.
Yes, please.
So as you mentioned that there is a slight change or alteration in the chemical structure of the drug, it can ultimately change the physiological or pharmacological activity of drug. So if we use certain drug delivery systems, for example, we work with the delivery of drug with nanoparticles such as metal-based nanoparticle or polymer-based nanoparticle. Do you think that using different kind of delivery methods can alter the structure of the drug, which ultimately leads to the pharmacological activity?
The system can change. So gold nanoparticle, silver nanoparticle, nickel nanoparticle, this one nanoparticle. What happens? They make the compound more soluble, more efficiency, and in case of nanoparticle, the dose is required lower. It's dose. Anything excessive drug. Okay, if it holds less, less toxic. Then particle cannot change the formula, but it can enhance the potency by the energy. Then particle is cardinal particle, gold is not particle. Anyway, so okay. Who gives you money? Thank you so much. Yes, sir.
For the last 30 years, there are more than 200 researchers on phytochemicals and nanoparticles for evaluation of their antidiabetic potential. But none of them are commercialized still there. Is there any specific reason? Because many of the research articles we are studying are based on nanoparticles or phytochemicals.
So the problem here, the third world country or developing country, we are academician. We are doing our research for our degree. Somehow you need the degree. After degree, you disappear. So normally during your MPhil or during your PhD, you are doing in lab work and you are maintaining a notebook, right? So notebook is not your asset. Remember this thing. Notebook is not your asset. It is the institute asset. And I did PhD in Japan. So notebook, it should be open. It should be in my desk. So your professor, your guide, anytime, what I'm doing, anytime he can check it. So the rules is there. So every time I work there in Japan, at the end, when I come back home, I have to do photocopy of my notebook and give it my supervisor letterbox. In the morning, okay, you see my progress. So even at night, he normally, if in their mind anything, immediate delay, will come to the lab. They'll read it and maybe think what this is the result, what is previous result. Here, my notebook is available for you. Notebook available, I have got a photo with him. So notebook maintaining, I learned that. That is how to write the notebook and how to maintain it. But in Pakistan, Bangladesh, notebook you write in such way that after 1 year or 6 months, you cannot understand by yourself. So it's a duplicate. The important thing is that, so I was telling you who told the Japanese. Okay, Japanese, I went there many times. I did many things I learned. So during marriage ceremony, right, we purchase lehenga, sari, you will use it only 1 or 2 days in your life. Japanese people don't purchase it. They have rent it. Number 1. Number 2, so you are cooking biryani, this, now. Your mother cook it, you see it, you get married, then after marriage, everybody is panic. मेरे बच्ची हैं कभी cooking नहीं किया, kitchen पे नहीं है, तो क्या करेगा? परेशान. Their mother, they have everything written in cupboard. How much rice is there, how much chini, sugar, and everything written form. And they have a written manual of cooking biryani. Purchase rice, basmati rice 1 kilo. Put in water, half liter. 5 minutes later, take the rice, stir it. Okay, make this kind of bowl there. Add water. When it's boiling, add this. 5 minutes, this one. In such way there, your mother and father, sometimes your mother is in hospital. I was in hospital. With the mother and the king, मेरे बच्चे की खाएगा. Extraordinary education. They make a guideline that you are anywhere in the world, so your son or daughter can follow the notebook, how to cook biryani, how to cook this. And during getting married, this is the best gift for a daughter. It's what we got to do. I hear how to do. Ammi, biryani pakana hai. Patao. Ammi hospital पे. Ammi का he is ever dead. तुम्हारे दिमाग में घुर रहा है because your husband asked biryani. So they said हमारे शो अलग. You are spending more money for 1 day, but your whole life you need the cooking materials, cooking manuals. So some way of thinking is also different. Change. Okay. Normally your background of science background, right? I am become science background. In my home, I have a fridge, refrigerator. Okay, in front of refrigerator. So during Qurbani, so I have meat, 8 packets. 1, 2, 3, 4, 5, 6, 7, 8. So when you 8, when I take number 1, we cut it. So inside the fridge, what is there? I know what is still top of the fridge door. I can understand. Some people open the door. Chicken kidhar hai, idhar hai. So use science in your practical life, in your real life. It will save your energy, save money. Okay, make your conflict with keep there. Postpone. It's for this. Make it numbering. Make numbering. Make notebook. Change yourself. Upgrade yourself. Change your tune. Then change for the Pakistan. Introduce something new at your home. Science doesn't mean you have to be only in the laboratory. Bench to bedside, bench to body, and bench to your family. Use science in scientific way to your family, to your society. Yes, sir. So this is the end. If you have question, tomorrow, right? Like that.